Okay, so we're recording, so we'll get started. So this is our first lecture. So we're gonna do introduction to bioinformatics and computational genomics, and Unix command line and high performance computing. So this will be our start. Okay. So I would like to start and say that in general, the data is taking the central stage across the domains, regardless what the domains are. So if you think about obviously genomics, genetics, the data is there, and the data is coming from the machines which are generating the sequencing data. So having the skills to analyze the data is essential to do computational genomics. But it's even happening in other domains like pharmacy research or pharmaceutical sciences, which I'm part of. So I'm a part of School of Pharmacy at UC, where we see the trend of quite of changes in the domain where the data, so where there is a need to integrate big data computational skills into education. And then it comes that there is a great opportunities for the research, bringing the data and analyzing the data, as well as facilitating reproducibility and transparency, meaning that all the details of the analysis is shared, right? So as you do research, unfortunately, traditionally is that people do the analysis and they share some parts of the analysis. So they don't share everything because it's challenging. You need to and you're describing that by words and you should give certain commands. So that's not a great idea. So the recent trend is that if you're doing code-based analysis, meaning you do analysis using the code, and if you do so, for example, that's Python or R, there's a tremendous opportunity to share that in the reproducible way, meaning that you can create what we call notebook. And that's quite a new term. I think it's around for 5 or so years, where you put together the code and the data and the results, and you can share that with everybody. And this is what we're gonna do. And this is what I think the best way to learn coding and more so data analysis using the notebooks. So we're gonna use Jupyter Notebooks, which is again, the latest thing I know. And that's really facilitates reproducibility, meaning that everybody can take your notebook and reproduce it. So that's important. And that's also transparent, right? You're sharing all the results. And then I think that brings this point about the crisis, reproducibility crisis happening at some level, at some scale. We don't know exactly the scale, but there is some crisis happening. And I think it's extremely hard in the wet lab, right? If you're doing experiment, it's very hard to document what exactly you do, like how much reagent do you take, which exactly the settings do you have, if you have a different lab settings, can you reproduce it, right? So it's all open question. However, I would argue that in computational science, it's more straightforward. If you have a data as the input and you share all the code and the commands you have run, I pretty much kind of optimistic that it can be exactly reproduced. You just need to share that correctly. And this is what we're gonna learn here. As you do the analysis, it's not only about the analysis, but it's also about how do you share that. And you can see that it's kind of become a standard in the field. So some journals are extremely strict about that. If you don't do that analysis in a certain way, they will not accept your publication. It's quite rigorous. I mean, it's quite stringent, but I'm glad that's happening. So it's transforming the way people do the analysis. And by the way, if you have questions, like feel free to, I think I want to keep it open. Don't keep your question to the end. You can ask them right away. Okay, so we will cover a lot. We'll talk about the advances in next generation sequencing. So what is the sequencing, right? Sequencing is studying the DNA of the organism, right? Like we try to sequence, we try to get the sequence of the entire genome. So if you think that, I mean, in ideal world, if the technologies are so advanced that you can just take the DNA and get the entire sequence of it, I mean, it would be no bioinformatics kind of, or at least probably will not have this lecture today, right? Because it's trivial, right? Like you just take the entire DNA, you save it to a TXT file or something, and you look manually. And this is what people did, like very, very, very early stage publication about human genome. They just publish the sequence. They say, oh, that's great. We discovered chromosome 19. Let's put that as a figure, right? Not anymore, right? That becomes like a common thing. So So, but then it was like that. People almost like semi-manually. And this is the plot, I think, the most motivating plot to me about bioinformatics. So what this plot is, it's about the cost, how much it costs to get human genome. So you can see that in 2002, that was quite a bit of money. And this is what actually Human Genome Project started, right? It was an international consortium. So people get together and said, hey, we really need human genome. Like we can no longer do individual genes in a kind of ad hoc way. We really need something systematic. We want to transform the field. And this is what the case, like really getting one reference, human genome, transformed the entire field and enabled computational genomics. So it was international consortium, many, many dollars being spent. But from that time, and then it was actually semi-manual, meaning that people generated the sequences, then they did like get the reads. So we'll get to that, but the reads are pieces of DNA you're getting from the machine, right? So, and then they kind of did some software. They also did manually. So it was like a lot of work. And the goal was just to get one human genome, like literally one genome. So it costed a lot of $1 million. Now, from that time, the biotechnology, I would say, community was working a lot to bring the cost down. And then it was successful. So usually in computer science, we say that the good trend is the inverse Moore law, meaning that the computers are in general twice cheaper every year, right? That's exponential kind of decrease. The same we have for COVID, that's exponential increase, right, of the cases. So this is exponential decrease. So that's a state of the art, right? That's the best we can do usually in technology. We can double twice, I mean, we can reduce twice the cost. And we are happy about that. That's great. Computers are evolving, a lot of computational power, great. But sequencing represents something super exponential, right? Something like outstanding, I would say. Like we managed to bring the cost down to the couple of $100 and then still go down. So $300 versus 100 millions, right? Like quite a change. So what that means? That means that the people are using it, right? If you're studying something, instead of old-fashioned technology, you say, why don't I sequence this? Why don't I discover new genome of some animal? Or why don't I apply genomics in studying some disease or microbial communities and so on? So that has transformed the science. Now, so what is required? So what is actually the compromise? I think there is always the compromises in life and in science. So the compromise is technology is a little bit worse than it used to be for $100 million, right? So again, we cannot sequence the entire DNA as one thing. We're getting short fragments. So think about the very, very long string of 3 billion, right? 3 billion, right? So 3 billion sequences, right? So this is what human genome is, 3.3. So, and then you are getting very short fragments, like very short sequences of 100, 300, around that. So now you need to restore the original sequence, right? So how would you do that? So that's why having human reference was very important, because there is a very simple observation that we are very similar to each other, right? We are the species, like the humans, we are 1% or less this like different. That's it, right? That's what determines you as you, less than 1% of bases in your genome. The rest is the same. Now, what that means technologically? And people did a very nice observation that you can use somebody else's reference genome and match your own reads from the sequencing machines to that reference genome, and then modify that reference genome. So this is how you can get your personal genome. So that's possible. And we will cover that. However, if you don't have reference genome, reconstructing your personal genome is impossible. And the reason it's impossible, just because you have repeats, as you might know, right? So you have regions in your genome which are exactly the same. And those regions are longer than your sequencing, like the reads you're getting from the machine. Meaning, no matter how hard you work from bioinformatics, you can be brilliant bioinformatician, you can be Nobel winning prize mathematician, you will not be able to reconstruct human genome. And I think this is really bioinformatics. It's not about only one domain. You know, if you're computer sciences with limited knowledge of genomics, or biology, I would say you will be less successful if you're balancing two fields. I think you really need to balance and biology and mathematics perhaps and computer science. And this is the good example. People come up kind of with the trick to reconstruct human genome rather than pushing the computational agenda all the way. And then I think it speaks to the field that it's really, really interdisciplinary. Okay, so this is how I see the field, right? So we have a lot of technologies. So we'll cover in this course 2 of them. We'll cover genome technology. We'll talk about whole genome sequencing technology when you sequence a particular genome of a particular individual, and you're trying to reconstruct a personal genome of that particular individual using the reference which we paid for, right? Which we paid as the global community already. And we'll talk about transcriptome. It's slightly different, right? We want also to study gene expression levels, let's say. And traditionally that was studies from expression arrays, right? And it still is in some degree, but the newer technology is to use sequences. So we'll have 2 applications as the module 5 and module 6. So in general, those technology generates tremendous amount of the data per individuals, and especially if you're doing a lot of studies. So this is really big data. I would say it's like big omics. By omics, we mean just all technologies, right? That's like the acronym. Genomics, epigenomics, transcriptomics, proteomics, you name it. So that refers as omics. So there is a big omics data around. And then that's on the left. On the right, we need to push that technology into clinical settings, right? We really want to like sequence somebody and inform the decisions the clinicians and the medical people are taking. But we can't just take the raw data and make those decisions, obviously. We need some frontier which will convert the very large and actually noisy. That's another thing I didn't mention. There is also those segments we're getting from the machines. They are not just short, but they are noisy to some degree. There is 0.1% errors being more or less equally distributed across the data, right? So you're dealing with partial information, but also noisy information. This is what makes bioinformatics hard, right? You have both. Now you want to study rare variants, that's very hard. You want to study, so that's I think one reason, like 0.1% error, that's one reason bioinformatics yet to be widely applied in the clinical settings, right? We really want to be extremely confident if we call something a mutation or something a variant, we really want to be confident that's really a variant rather than a false positive. So that's why we need bioinformatics. Like we need to have a frontier which will take care of those, right? You as the clinician or somebody as the clinician doesn't need to, I mean, you need to know, I would say in general, and this is what this course will be also about, like you want to know in general, what are the limitations of bioinformatics and what is kind of assumptions and trade-offs bioinformatics tools are making, because they do so. But you don't need to kind of exactly know, but on the high level, but bioinformatics take care of those things, right? It takes the data which is noisy and partial and derives as accurate as possible interpretation of the data, right? Which can go to the clinical decision making. Okay, so big omics data, right? So this is what we'll be talking in the module 5 and 6 as we are prepared. But I just wanted to kind of jump ahead and kind of give you a big picture. And this is what today kind of, I would say, half of the lecture will be about big picture. So some things might be not clear to you yet. Stay with me. Hopefully they will become clear towards the end of the course in January. But the second part of today's lecture will be very hands-on. So we'll cover the particular things which you will have kind of chance to start practicing things. But this is about big picture. So yeah, so by big data, we just mean big data, massive amount of the data sets, which has tremendous opportunity to be analyzed, right? And then comes with the challenge, being big, being noisy, being partial, and so on, unstructured, and many kind of typical challenges with the big data. And I think one important thing I would like to emphasize that, which I will point you to a couple of publications we pursue. So my lab, for example, is purely computational, right? We don't have any like labs, wet lab component, at least in my lab. I do collaborations, but I don't have personally per se like a wet lab component. And this is what we actually envision for the computational medicine and bioinformatics laboratory at your university, right? We don't envision yet, at least, to build like facilities because it's challenging and it's costly, but rather try to be what we call dry lab. Dry lab meaning that we do everything computational. So the model is that as you saw previously, the sequencing cost and the cost of the technology in general drop down. So what that means? People generate a lot of data. And the current model, luckily for us, is that as you publish your data, you need to share that in the community, which was actually, by the way, a great idea to do that. Now, what that means, we accumulated quite a bit of data, so we can reuse that data, right? We don't need to generate new data. We can rather do some sort of meta-analysis, meaning combining multiple data set, as well as we can like repurpose the technologies, which I will also talk a little bit about. And then we can do novel biological and translational clinical discovering using existing data. Meaning, actually, I would argue that's a great model for the countries with limited resources, like Moldova is, right? I would say that's challenging and hard to build state-of-the-art facilities like wet labs. But doing this secondary analysis, I think is doable. All you need is knowledge. And computational resources, there are around. Like Amazon is giving free credit, Google Cloud giving free credit. There is resources. I would say knowledge is the key. Knowledge is like what makes it happen. Okay, so when we say biomedical data science, that's kind of the definition I see. Biomedical data science, interdisciplinary field that combines computer science and biology to research, analyze, and interpret large-scale biomedical data. So what we mean large scale? Many samples of data points, right? Many individuals. When we do the study, we want to probe many individuals. Meaning that you need, if you have the algorithm to analyze the data, you just need to repeat it many times. Let's say you have 1 million patients, you need to repeat it million times. Or you have million data points, repeat millions of times. And the data is not simple, right? It's complex. So it's large and requires code-based solution to process and interpret and visualize that. So we'll here focus on the code-based solution. So as the example, I will be giving you very simple files, simple ones, and you will feel that, oh, I can do that in Excel for now. Yes, for now you will be good. But as the file size grows, and I don't know out of my head what is like the limit, but I think like 10 million perhaps or something, if you have a file of 10 million, I don't think you will be comfortable analyzing that in Excel. But also, very important, it's not scalable, I would argue. Excel is not scalable. You can do one sample, perhaps spend a couple of days. But if you need to do 100 samples, would you spend 100 days? Probably not the best investment of your time, right? Rather, you want to develop code-based solution for one sample or one data point and like analyze that and then just scale it. So scalability is the key. Scalability is extremely simple when you know coding, right? You can scale. It doesn't matter for you. Do you analyze 100 or 10 samples? It's the same. It's exactly the same, just like runs more. The computer runs, but it's not your time. You can go get the coffee, enjoy your life, come back, and the analysis is done. Somebody asks you to adjust parameters, right? Like you do the analysis, it's very often. You go and you do the analysis and you discuss with the colleagues, and it's not the best one. Say, no, we need to adjust here and here and here and here. If you do manually, you spend exactly the same amount of time, right? But if you're smart and you know code-based, you can just alter certain parameters, click the button or like whatever, run a couple of commands, rerun the analysis for a day, enjoy your life, come back, analysis is done, and present the results again. And do it as many times as you would like. I think that improves rigor. You're not afraid to explore parameters, which you often are if you do manual, because you don't want to spend, again, such amount of time, right? So you're more rigorous. The analysis is more robust. So I think it's a good thing overall. Okay. And then another thing to mention that what is specific about biomedical data science, the algorithms which are analyzing, well, like bioinformatics, I would say in general, bioinformatics, biomedical data science, the algorithms which are analyzing raw data from the sequencing machines requires a lot of computational power. So typically, you cannot run them on the laptop or whatever the computer you have. You just can't. They require more. But more so, you are dealing with many patients. Even 50 patients, it's hard because, like, let's say you can do it on your laptop, one sample, but you need to do consecutively, right? Like one by one. So 50, let's say one analysis takes 1 day. So 50 days, no, not a good idea. But that's why we're using what is called high performance clusters. I mean, in this course, unfortunately, I will be not yet able to get you the access, but look, we can, like, if you really need one towards the end, we can discuss it. But obviously, we'll get access for those of you who will be employed in the laboratory and will participate in real research. Then you will get the access. So kind of we're using here the proxy. You will get the sense, but like unfortunately, we will not get like the access to the high performance cluster. But this is where we do real science or like real analysis. Because again, that's the only way to analyze, to run bioinformatics tools, because then, or the cloud, usually the cloud or the high performance cluster. And then it's scalable, right? You can submit 50 jobs, meaning you can submit 50 different analyses in parallel. So if it takes a day, that 50 takes a day, because cluster has the capacity to run 50 things at the same time. So it's scalable, it's fast. So you can run many things and be effective. Okay, so I think that's how I see biomedical data science. Even if you don't plan to do big kind of data analysis, I think the course is still relevant to you, just because even with the small data, code-based analysis allows reproducibility. And again, I would argue it's more effective because you can repeat and explore different parameter threshold, change things in a very effective way, right? Okay, so I mentioned that sequencing became cheap, right? Or relatively cheap. So it's accessible. And actually, by the way, since it's accessible, I think the plan for launching bioinformatics in Moldova will be to do what we call Moldova Genome Project, right? Actually, our neighbors in Ukraine, they already did it. So what they did, they took 100 samples across Ukraine and then sequenced them with the whole genome sequencing. And then they discovered quite many variants which are unique to Ukraine, as well as things, structural variants like deletion, insertions, translocation, so on. So I think it would be nice to do it in Moldova. And that's, I think we would need somebody to analyze it and so on. So that will be one exciting project. And again, sequencing, it's accessible, you know, like it's expensive, but I think as we think about the country level project, that's an okay price. Okay, so but what that means that we brought the sequencing cost down, that means that we have accumulated the data, right? Remember, I said that as you sequence in your wet lab and as you publish, you will be required to share the data. And there is two major repositories, like locations, where you can upload the data. So one is GEO, Gene Expression Omnibus, and another is SRA, Sequence Read Archive. So two of them. So traditionally, GEO was more for gene expression data, and traditionally it started for gene expression arrays. Right now, it's also accept RNA sequencing. Then SRA, it's for all sorts of sequencing, like everything, whatever relates to sequencing is called, I think it's Sequencing Read Archive. So this is what it stands for. Yeah, it's written there, Sequence Read Archive. This is what it stands for. So any sort of sequencing data in the world will go there. So there is 5.7 million samples there, quite a lot. So for example, I can tell you there is 70,000 RNA-seq samples being there. So I think that's tremendous. That is a tremendous. I see the hand from. Is it Verial? Is it you or somebody else? No hand. There is a hand. So I mean, maybe I don't see everybody. So if you want to ask questions, just speak up rather than raising the hand. Okay, yeah, you can like ask the question during this thing. It's not like monologue, so you can ask questions. Okay, so. Yeah, so like it's a tremendous resource. And then I would argue it's barely touched for various reasons. It's actually a little bit noisy, right? So as people share the data, they forgot to share metadata, for example. They forgot to say, like, for example, they did the study of sepsis, let's say, right? And then they did whatever they wanted to do, discover some biomarkers of severe sepsis, let's say. But as they upload the data, let's say they upload all raw data very diligently, good for them. But they forgot to share metadata, or they partially share metadata. For example, they say, okay, we'll share biological sex, we'll share age, we'll share something else, but we forgot to share mortality, who died and who did not die, and so on. So the data, I would argue, there is no point in this raw data right? Like, yeah, great, I have raw data, but if I don't have mortality, I cannot run my models, right? I don't know who survived. I cannot predict it because I cannot validate it. That's it, it's useless. So there is a lot of issues like that. I would argue some phenotypes you can restore, like biological sex, okay, not a big deal. You can just look at the special chromosomes and you will get it, right? It's possible. Age, harder. You can get some proxy of the age, but not that accurate. So age is required, and so on. Ancestry, like Europeans, Asians, which country, you can partially do. I mean, from whole genome data, you can do very accurately, actually. From RNA-seq, that's a research question we are exploring. Can you infer ancestry from transcriptomic data, right? And things like that. So there is a limited accuracy, like a limited completeness of the public data, and two is very large. You need a lot of kind of effort, but also cleaning the data. So not that many people do that. But again, to me, this is how I envision the laboratory to work. It will be analyzing public data. It will be like, there is actually, if you want to study a particular disease, for very many diseases, there is enough data being there. So you can discover biomarkers just purely from the public data. And then people did quite a high caliber of discoveries publishing in Nature and other journals just from I think it was tuberculosis. They found, I forgot the cell type, but they detect the cell type from like cell type composition from transcriptomics data, and then they conclude, I forgot the name of the, let's say, macrophages or something like that, neutrophils, I don't remember exactly, but those were predictive of the mortality from tuberculosis. It's a big deal. So that is like a big deal. So that's a high caliber discovery purely from the public data. Again, no wet lab component, zero data generation, meaning it's almost like zero resources, right? So this is the model which is suitable for countries, again, like limited resources. So that yet to happen, like that's not yet happening widely, and I hope it will happen in Moldova. And I think that's the only way for Moldova to jump into bioinformatics and produce some good caliber discoveries. From public data, combining maybe with the local data, sure, we can generate some local data combined with public data, and this is probably will be the best setup. Okay, coming back kind of to the data, right? So we have a lot of data. The genomic data is around. As we speak, it's free to use. You can go now if you have the skills, and if you have storage and the skills, you can download as we speak, right? So it's around. Just need to know how to analyze it. And this is what this course will be about. How can you use public data? How you can, not necessarily public, but how can you process genomics and transcriptomic data in addition to kind of more data science things. Okay. Okay, so this is what we call computational data-driven research. This is what my lab is doing, and I obviously bias about that, but I strongly believe that the dry lab research can contribute to science to the same degree as the wet lab. We just have a different kind of skills, kind of components, or like we have different kind of goals, but there's a lot of similarities. It's not that different as you think, right? You would think, okay, wet lab research, they recruit the patients, they sequence them, they study the particular disease, and the dry lab, you do very, very different things. Not really. We do quite similar things. In what sense? So that's just a different level, different way to get data, right? So to get data for the wet lab project, you need to recruit the patients, you need to get the samples, like biopsies from them, sequence them, or whatever you do with, and then get the data. And in the end, you kind of will have a CSV file or Excel file where the columns are the patients, the rows are the patients and the columns are the phenotypes, let's say mortality, age, and whatever, like genes, and so on. As dry lab, as computational, we will do it from the public data. So we cannot sequence, so we're gonna go for the public repositories, find all the reliable data sets which are coming from the particular disease. Again, let's say sepsis. And actually there are around 3,000 samples around as we speak. So we can take those samples, we'll have a table with 3,000 rows and some phenotypes. Let's say we have cell type composition, which we can infer computationally from transcriptomic data. So what we have age, biological sex, mortality, and fraction of the B cells, fraction of the T cells, fraction of macrophages, fraction of neutrophils, and mast cells, whatever, so on. So this is our table. So at that point, in the wet lab, if you need that, you would run, let's say, flow cytometry, right? You need cell type composition, you will go to the wet lab, get access, let's say, to the machine flow cytometry, get your cell type composition. So in the stage where we kind of have a summary data, it will be the same, just the way we got the data is different. And there is obviously limitations and advantages. Limitation of lab, you can recruit the patient you would like, right? With the certain phenotypes, maybe severity of the disease, you can control it. As we do the public data, we don't really, we work whatever is available. So if there is no data for the particular disease, we cannot study this disease. I think that will go away as more data is generated, right? My assumption is as more data is generated, like we'll have more and more diseases being covered. So that will go away, but we cannot control so on. So that's a plus and the minus. The minus, yeah, we cannot really like design our experiment, but the plus is that we are getting really heterogeneous data, right? Like real world heterogeneity. People are different, different ancestry groups, people have different symptoms, geographical and so on. If you just study, let's say if you study in Oklahoma, which is central, in the very center, or like Nebraska, in the center of US, majority of the people there are European based, right? They're white. So meaning that whatever you find can be only relevant to the white people, to the Europeans. That's it, right? If you take those biomarkers, go to Africa, they might not work. So that's a, and as the vet lab in Nebraska, you actually don't have another patients or very little, let's say, African Americans, very little Asians. You cannot really capture them. So you're limited. So if you're using public data, you can bring data across the globe and kind of get as much real heterogeneity as you can. It's challenging, but to deal with heterogeneity, but this is how I think it's quite a prominent approach. Okay, so this is like how kind of to give you the idea about the dry lab. Okay, any questions so far? Okay, so this is how kind of very simple schematics. What do we mean? So when we say that we reanalyze the data again, we say from public data, we call it secondary analysis. So I want to contrast that with the primary analysis, right? So primary analysis, meaning that you do in the vet lab. So if you do in the vet lab, you take the biological sample, you sequence that or whatever that you do the technology, you upload that, you share as you publish, you do some computational analysis if necessary, and you get the results and you publish them, right? That's kind of very simple primary analysis. What is secondary analysis? We skip first three steps, and we immediately start with the public data. We run computational analysis, we do the results, right? So like that's just to bring everybody on the same page. That's kind of oversimplified schematics. So we'll be focusing obviously on the secondary analysis, right? We'll not cover anything wet lab. We'll talk a little bit about technologies so that you know, because so you know what you're dealing with, but we'll talk about computational analysis required for the genomics data. Okay, to give you some motivation. So if you compare, and this is actually the project we are working in the lab, and this is some kind of the project you can participate theoretically, like giving the skills you have from the class. So we were curious, right? So since we know that there is secondary and the primary analysis, so usually in the research, we try to take the question all the way, right? It's not kind of enough for us to say, yeah, there is both. There is some people do primary and secondary. I really want to know the numbers, right? I think this is what we can contribute as the bioinformatician and data scientists. We can really put the numbers on things. We can quantify things which are happening in the world. And this is what it is, right? Like if you ask the question, what is the fraction of the papers doing secondary analysis versus primary analysis? Like what is there, right? And actually nobody knows the answer. So you're probably like one of the few people in the world who can see those numbers, and we hope to publish this soon. So we'll share in the world, but for now we did not. So what did we do? So actually, as we speak, all the publications from the PubMed, right, like the repository where there is literature for medicine and biomedical research, many of them, some of them are restricted, but majority are publicly available. So you can go ahead and download all of them. So you'll get around like 5 million, maybe 3 million files. So that's possible. We did it. So we take all the public publication, the publicly available publications. Okay, so we kind of claim we have all the literature in the world sitting on our cluster. Great, so what next? So we said how we can differentiate what is secondary versus primary. We thought maybe they say it, we check, no, they don't. There is no way automatically, unless you read the paper manually, but we didn't want to read 5 million papers, right? It will take us forever. So that's not good. That will not work. Like, how else, right? How else can you differentiate secondary versus primary? We thought, okay, actually they mention, luckily for us, they mention IDs. If they generate the data and share it, or if they reuse the data, they will mention SRA ID, and it will be like letters SRA and 6 digits. Like really, they will say it in the paper. Unfortunately, they don't say it exactly in the same place. You know, sometimes they have data availability section, sometimes they don't, sometimes, you know, like there is a mess. And sometimes they say we generated the data, sometimes they don't. So it's impossible to differentiate. But we have those now, we have those IDs. We said, forget about the structure of the paper. You know, like it doesn't matter. Let's just extract those IDs. Since we know exactly what they are, let's just extract them. So now we have all the papers and data sets corresponding to those papers. We had, okay, that's a good start. Like, but how can we know that who did secondary versus primary? So we did some assumption, which I think is quite a valid one. We assumed that we have all the literature in the world, which I think is valid. And we said whoever first mentioned the data set, that person of that paper is the paper doing primary analysis. And if somebody else mentioning this data sets after that paper, they're actually reusing it. That's it. That's the idea, right? So now we can classify every single paper and the secondary and the primary analysis. And this is the numbers across all the years. So you can see that for SRA, there is majority doing primary analysis, and there is some fraction around 20%, even more, doing secondary. Not bad, right? 25 I think is 25 I think is a good number. But in geo, that's remarkable. There are more papers doing secondary analysis than the primary. And again, I don't think anybody knows about that. So to me, it's remarkable. People doing more secondary analysis. And the field is new, you know, it's like around 15 years. Like what is like we're doing. So we have like people who are doing dry lab research are more popular, you know, like that's a more popular approach. And there are many reasons for that, because it's more scalable, more appealing, less resources, and so on. There is a lot of drawbacks, meaning that you should trust the people who do secondary analysis. It can be like fast and dirty, you know, like, so we don't know that. That's one limitation. And then you should, like, you know, do quality controls and so on. So, and actually I will point you to the paper we are writing about kind of challenges of secondary analysis. And what we did, we interview all the best, not the best, but like high level, let's say, people across the globe who are leaders of the field, what they see challenges of secondary analysis is, and we kind of summarize it. And I think one challenge was that computational people might have limited background when they analyze the data and so on. So, but this is the numbers, you know, like here we were really interested in the numbers. Okay, and this is the trends across the years. Again, that's just to motivate you. So for SRA, you can see things did not change. On the left, SRA, things did not change much during like across the years. As for geo, there's a clear trend. There is more paper doing secondary analysis than the primary, right? More and more. To the degree that 2016 was a transformative year where it became more papers doing secondary analysis versus primary. And this is the bars, every bar corresponds only for that year. So those are not cumulative. Okay, so that's I think encouraging. So you're in the right place to get something that many people use. Okay, so in terms of are we utilizing all the data? No, we are far away from it, right? So there is at least 60% of the geo data sets never being touched for second time. And there is even more data sets never being touched again for SRA. So why it's a big deal? Because actually it's a big amount of money people spend to store those data sets. And the idea is to store them for generations, meaning, you know, like to preserve the data. But there is a little effort being put to make them reusable because they are large, right? And then that's challenging. But this is how things are. So any questions? And also, if you think about like superstars, meaning the data sets which are constantly being reused, So you can see here that some data sets are used hundreds of times. Like why is that? We don't know yet exactly, but I think it comes with the disease. For example, the highest reused data sets in gene expression, it's cancer. Certain cancer cell lines, they're constantly reused. For SRA, it's less clear. The top were cotton somehow, like we don't know why. But we're still exploring what makes the data sets usable, and what we see preliminary results are like the sample size. So if the data is sample size, meaning number of samples in your study, right? Sample size. So if number of samples less than 10, like roughly speaking, nobody cares. Nobody will reuse the data. It's too little. So more than 10 people will use. So sample size is driving the reuse. But there are other factors which we're exploring. Okay, so conclusion about reusability. So reusability of omics data varies across different repositories, right? As you can see, it's SRA versus Geo. And we think the reason is the Geo is more reused because it's easier. So roughly speaking, what is the Geo data? It's just a big matrix or like a big table. Every row is the gene, or let's say every row is a sample. Let's say you have 100 samples, so that's 100 rows. And every column is the gene. So we have like around 60,000, the table we can manage kind of in Excel. So I think that's one reason. And sequencing data is nightmare. Like you should know bioinformatics, you should have access to the cluster, you should rerun and so on. That's like hard. So that's one reason people don't reuse, or they cannot reuse SRA data much. So this is where we are. I just wanted to bring you to them kind of, to bring you the knowledge about the field. Like this is where we are. Okay, and this is like another kind of, just want to mention briefly, like this is another reason to reuse the data. Like for example, if you're doing gene expression and you're using RNA-seq, great, everybody does that. So let's say you did, I don't know, like particular disease against sepsis, and you did 1,000 samples. You did it, you discovered some genes being responsible or whatever, having predictive power to sepsis, and you uploaded data, you shared with the world. Now, like my lab, let's say, or you are saying, I want to reuse this data. So what you can do, you can look again at the data and extract something else. You can repurpose the technology. Like originally, technology was designed to study gene expression, but you say, you know what, I want something else to extract. And you can, for example, you can, among cells, right, in your samples, it depends on the tissue, but there are B and T cells floating around, right? They are there. So now they have their own genomes, every one of them, right? And then they have VDJ recombinations. They have a recombination of certain genes, which are producing the novel genomes. So as you sequence, you kind of randomly will take some of them and you will sequence them. So now you have some evidence about those recombinations. So you can, and this is what we did the method for, you can reconstruct VDJ recombination and clonotypes, like CDR-free sequences. And you can quantify the immune response. You can say there's a high immune diversity, low immune diversity in the T cell, B cells, alpha chain, heavy chains, like whatever, all the biology. And you do it from the RNA-seq data. So now, and you also can do what is called cell type composition. So what is called deconvolution. You can deconvolute in silico, like computationally cell types. Now you know exactly what is the fraction of every cell type. Now we are talking about some new data, right? So when you study sepsis, you say, that's great that they have discovered 1,000 genes, let's say, contributing to the predicting of severity of sepsis. But 1,000 genes is a lot, right? It doesn't say you much about biological mechanism. I mean, you can do pathway analysis, you can look at the cluster, the function, but still it's 1,000 genes, like a lot. You didn't narrow down the mechanism closely. But if you say that the T cells are like the fraction of the T cells, or let's say T cell receptors are predicting survival in sepsis, that's another story, right? Like you kind of pinpoint the mechanism and you can further study in the wet lab, but that's important discovery. So this is how you repurpose, right? You look beyond what the technology was designed for, and luckily bioinformatics offer that. So this is all the things you can do. You can do microbial communities. if you have enough microbes in the sequencing sample, there is challenges for that, but you can theoretically do that. So just wanted to give you kind of like the reasons for repurpose.
Sergey,
Okay.
what do you
Yeah.
imply by data convolution or?
In real tools, we actually run benchmarking. So there is around 11 tools which are taking gene expression data and some reference, which is publicly available, and gives you back cell type composition. And there's 11 tools, and I think we publish like this year in briefings bioinformatics, and we detected I think 2 tools, Get and CyberSort, are giving the best results quite reliably.
Okay.
Because what we did, we compared that flow cytometry. So we got 3,000 samples where we have both RNA-seq, no, sorry, it was gene expression array and flow cytometry. So we treat flow cytometry as the truth, and we compare, and and then it was pretty reliable. Cool. Any other questions? I think we can skip this for now. Okay, so a couple of more slides, and probably we'll take like a small short break, and we'll kind of get to the more hands-on things. Okay, so another thing, we will talk a lot about transparency in this course and reproducibility. Again, sharing everything, how you do the analysis, so other can exactly repeat your analysis, right? But it's not only about others; it's actually about you. Like you think about, yeah, I should do it for the community, I should do it for others. No, you're doing that for yourself, because you will be more effective researcher. Meaning that, like, think about science, right? We submit the paper, and if, like, the best case scenario, of course, paper is reviewed, but like in 3 months, you get the report back, and they ask you to do something. Do you remember anything? Personally, I don't in 3 months. So it's very hard for yourself to revisit everything, to rerun. But if you structure everything nicely, one, your collaborators can help you because they can rerun the code and can help. You as yourself, as time go on, you can come back to the project and easily rerun them and adjust them as needed, right? But even in real time, that gives you the framework to try various parameters and to try to do things. Like, rather than, again, being afraid to do it manually because it's so laborious and takes so much time. So that's good for the community. That's good for yourself. Yeah, so like reuse the code with your laboratory, with your people around you, and for the future self. I think both. So this is kind of very typical statements from the papers, and I think it was Nature Communication, maybe Nature. People say how data is available and how code available. So data availability is simple. Just upload the code. I'm sorry, you upload the data, right? And you give unique IDs. So this is the IDs I was talking about when I said that we identify those from the paper. Remember? So those are here. EGA, AS00, whatever, this thing. It's not SRA, but it's European genomic something. So that's like, there are a couple of things, not just SRA. So we were extracting those actually from the papers, like automatically. Actually using the tool called grep, which we'll cover today. So theoretically, you can do the same. Okay, so that's more or less simple. So code availability is harder. You mentioned all the tools you're using, and also you're saying where is your code. But beyond that, if you have a box plot or the figure, you should provide the data points of that box plot. And as you know, the box plot doesn't convey you the data points. And probably you've seen that, like you can have different distribution, but the same box plot, right? So box plots is limited how it presents information. So you need to give data points. In ideal world, and we'll cover that, I think you should plot data points in addition to the box plot. I believe so. And actually, not me, but Nature journals as well. They require that. You cannot publish just box plot. You should put data points. So this is the code availability stat. So we'll be able to produce it. So we will have enough knowledge to write these kind of statements, which are required. If you want to publish high-level journals, you should. Okay, so example, right? Data availability. All data required to produce the figure and analysis performed this paper are freely available. This is our paper, IMREP publication. And we even say the exact panels, the exact figures you need the data for. So everything is really reproducible. Okay, code, yeah, so like this, right? Okay, so what is other requirements? So this was talking about the box plot. Box plot is great. We'll learn how to do that, but we also learn how to put data points. I think it's informative. It doesn't hurt, right? It kind of tells you a little about the data. Like you can appreciate not exactly, but approximately how many data points are per box plot. And actually, if you want exactly, I think it's helpful to put the number. If you pay attention here to the panel A, you can see that on the x-axis here, you can see the sample size per box plot, 211, 215, 93, 71, and so on, right? So I think that's good style. We will learn how to do it. I think N is a little bit hard. I don't think you can do it automatically. You would need to edit in some later editing of the paper of the figure, but data points and the transparent box plots we'll learn how to do. So I would say that's state of the art, like what you see here. It's state of the art way to visualize the data. And again, judging by that's like something like Nature or Nature, Cancer Nature, something like Good Journal. So we will learn that. So we'll learn how to produce these kind of figures, right? That's one goal besides bioinformatics. Okay, so this is we heard. So this is okay. What are the approaches, kind of to summarize? What is the approach to do secondary analysis? So first, you need to access public data. You need to identify data of interest based on the phenotype. Let's say you're studying all the people with certain disease, people of certain ancestry, whatever the phenotype is. Learn how to download the data from GeoNSRA, run bioinformatics methods, which we'll learn here, and repurpose if necessary. Repurpose, but that's just another way to say that you will run specific bioinformatics methods. Okay, what are the barriers? It's not only opportunities, there is a lot of barriers to overcome, otherwise everybody would be doing it, right? So lack of computational capacity, you just don't know how to do it. You're in the right place, we'll cover this. And the same of like inability to perform bioinformatics analysis in terms of infrastructure. That's challenging again. So as we move forward, we'll try to get you the access, especially if you will be done faster with individual project and you want to jump on the real project, we'll give you the access to the cluster. But that's challenge to some degree. We can also apply for the credit from Google, from Amazon. I think some of you did already. So that's possible to overcome. Control access to the certain data set. That's tough. If they don't want to share, it's hard. And like sometimes it just requires you to say, like to send one paragraph and say, hey, I want to use your data and give some valid reason, research reason, they will give it to you. It's okay, you can do it. Sometimes they almost require IRB, like internal revision board, like a formal process, like nightmare. We are dealing with one data set, it's just like a nightmare to get access, like half a year we are waiting. So that's tough. Sometimes they take it to the extreme, meaning they just don't want to share. And a good example is Qatar, the country, Qatar Genome Project. Basically, they said to access our data, you should come to Qatar, become resident, and then come to the special room and sit there in that room and analyze the data, right? So probably not many people will do it. So meaning that's another way to say, like, don't touch our data. But they didn't say that explicitly, but rather implicitly, right? So, and then incomplete metadata. That's something we do research about. It's hard. Again, as I mentioned, if you don't have disease or mortality, raw data is useless, right? You cannot train your models. You can do nothing. So that's a big challenge. So those kind of overall the challenges are. So how to overcome that? So inability to perform bioinformatics analysis, I would say we are doing this right now, right? We're empowering you with the right skills to do that. So that's fine. That's possible to do. I think I didn't have slides input. Also, the metadata is partially can be addressed by inferring. Genomics can be used to infer metadata like biological cells. I don't think there is a problem, right? You can just like infer it from the data. As I mentioned, age is harder. Other phenotypes are harder. Okay, so what else was there? Yeah, and control access, you can do nothing about that, right? It's just like, if they don't want to share, like Qatar, you will probably not get access to it. Okay, so this is how the overview, kind of like, I really wanted to start rather than jumping directly into like code and stuff. I just wanted to give you kind of high level bird view of the field. So you kind of appreciate how things are and where we are going. So now we'll go to Unix, which will be less exciting, a little bit boring in some sense, but but it's necessary. It's necessary to know that, to do analysis. So any questions for the first part? Okay, if no questions, so yeah, so let's get to more kind of practical stuff, right? So, I mean, I hope you got like a good introduction on overview on the high level of the field. So now let's work to get the skills, right? So I think it will be like this. So we'll spend quite a bit of time to go through technical kind of details and like through kind of any sorts of like working through and so on. And then I think towards the end, you will kind of get back to this kind of big picture, right? But now we need to focus on kind of details. Okay, so we'll go through the slides as much as we can today, right? So we have, I just emailed to TAs, so we'll have 2 students, which I will introduce you to, like they will join like in 45 minutes, right? So meanwhile, we'll go in 45 minutes as much as we can through the slides, and then the TAs will join and they will be helping you to kind of use the Google Collab, like they will go through again the commands, they will show you how to run them and so on, right? Okay, so what is Unix? So Unix is operating system very similar to the, I mean, not very similar, but like it's similar to the Windows, right? Windows as an operating system, Mac OS, Mac operating system, and Unix. So Unix was actually developed very long ago, and that's command line, meaning that you don't have a mouse, right, at all. Rather, you do everything through commands. And we would need that, especially for the bioinformatics component. But I think it's helpful to, in general, understand this interface because it's quite very robust. And you actually, all the analysis bioinformatics is done, my vast majority is done on Unix-based machine. And actually high performance cluster, like supercomputers, they are Unix-based because they rarely crash, they're reliable, they can be managed easily and so on. And I think data sets are large, as we discussed, right? The data sets, especially from the sequencing data, is large so that you don't have a choice. So usually the only way to do it is analyze on the Unix-based machine. And this is one example. Actually, Alexei, this is in Barcelona. I don't know if you have been there, but that's like, I took the image. I think it's pretty. They put
Yes, I know. It is very nice. They provided several opportunities to visit it for free.
Right. Yeah,
I don't know now during the COVID, but it's really great. It's very interesting.
No, I think it's nice. They put like inside some old buildings, like those machines. So that's cool. So,
yes, it is a church.
Oh yeah, okay. Actually, I don't remember I have been there, but like, yeah, I took this image. I think it was cool. Cool. Okay, so this is how it looks like. Those like Unix machines, like, you know, like a lot of computational power, which you can access remotely. And I think that's the key, right? You don't use your laptop to run any analysis. Rather, all you need, you have internet to kind of operate your part of the cluster, right? So that's important because what can happen, like it's actually convenient. You can run the analysis, close your laptop, you know, like go home and do stuff and come back in the morning, reconnect and check the analysis, right? It will be keep running there. It's stable, it's reliable. If you submit tomorrow, most probably you'll get result. If you submit, today, most probably you'll get the results tomorrow. It's very reliable. As your laptop can crash, you can close it and you can interpret and so on. So that's a reliable model. So you as the, let's say, biomedical researcher, do you need to become a programmer? I would say not, at least for this course. Of course, if you would like to, I'm not here to say that you should not change your career, but for this course, I don't envision you to become a programmer. Rather, I want you to get the essential knowledge in Python and bioinformatics and Unix so that you can utilize the internet. And there's a lot of resources. And I think Mihai mentioned Stack Overflow in the chat. So, you know, there's so many resources which are not available to you. And the reason that they are not available to you because you don't have the knowledge yet. But the goal is to get those knowledge so that you can read and utilize those resources. And you will see, after having some certain amount of knowledge, things will be quite easy, quite straightforward for you. So you can expand them. It's not that you take this course and you stop. Rather than, I really was hoping this course can be a start for you to explore data science and bioinformatics. Okay. So why not Excel? I think we mentioned this kind of through the course. I mean, for the lecture I was giving already, but I think it's good for the simple task and small data set, except gene names, as Alexei said. So that's not good. But Excel is not able to handle large files, right? So this is the limit, right? It's like those exactly the numbers. I think I put those numbers a couple of years ago or a year ago. Maybe they improved a little bit, but I don't expect them to improve dramatically. Like you're limited, right? You have limitation. Not talking about if you have multiple data sets, like would you run multiple Excel files, like, there is unclear what to do. Yeah, large number of data sets, not good. Not talking about reproducibility. Excel is not reproducible, right? You cannot, it's extremely hard to share the steps of your analysis, like how, like would you do a table with every step, but it's still subjective to human, right? To emphasize or like to enable reproducibility, you want to avoid human interaction, right? In the sense, code is unique, right? If you write the code, is uniquely interpreted. As when you write words and steps and tables, those can be misinterpreted. So if I would argue code-based is the better solution in general than kind of manual one for Excel. So why learn Unix? So because we start with Unix, right? Then we will transition to Python. So most new bioinformatics tools are created for Unix. So kind of you don't have a choice. Unix is very efficient, manage extremely huge amount of data very easy, very popular. So it's easy, relatively easy to get help and then find information, very stable, and the computers which are clusters which are on Unix never crash. And that's the reason Unix is behind high performance computer cluster, but also behind the cloud. So, like it's the same. As soon as you learn Unix, you can unlock opportunities to use high performance clusters as you have chance to, or if you have some credit on the Google Cloud or Amazon Cloud or whatever the resource. So Unix is there. It's kind of similar. So that's a fundamental thing. Okay, so. Okay, so Mihai, what did you say? This open source, as I know, what is open source?
Linux.
Ah, Linux. Yeah, it's open source. So you know exactly.
That is the beautiful thing. You can do anything that you want and it's secure.
Exactly. Yeah, that's a great point. Actually, just like I think it's like just like if you have comments, just speak up. I think because I don't have activated the window. I occasionally saw it, but like I don't see your messages right now because I'm in the presentation mode. So if you have comments, just like speak up. It's fine. Great. No, that's a great point, Mihai. Okay, cool. So we would like you to empower you with Unix literacy, right? And I think those two kind of what I mentioned. So we're writing this review. I mean, we wrote this review previously in Nature Biotech about like why bioinformatics and open data is a good way to boost science in countries like Moldova. So if you're interested, have a look. I think it's closed access. So if you're interested, send me the email. I can give you the PDF. You cannot find. I don't know if SciHub is still around. There is like a. Is it?
It wasn't updated for a year because we have lawsuits in India, but it's still all the data until that moment it's still around. So
I see. Okay, cool. Yeah, that's like I don't know if you don't know about SciHub, check it out. Read Wikipedia. It's quite interesting. Like one kind of lady, I would say in Kazakhstan, she's like transforming the science worldwide. That she's making all the closed access publication to be public access. Like she's just storing all the PDFs. So sometimes I'm just like, I even check that, but it's a little bit controversial. But
Robin Good of science.
Yeah, exactly. But it's interesting. Like one person can change the world to some degree. So which is fascinating to observe. Okay, so and another paper we are writing, and the preprint is available. Just like you can Google that name, Path to Independence. So we think people who do computational research, they gain independence and the voice in the biomedical research. And we overview the challenges and opportunities. And this is the paper I mentioned that we have interviewed many people in bioinformatics of high caliber, kind of to ask them what are the challenges and what are the opportunities and so on. So we will update that preprint, but there is some version online. So if you want to get some background, you can get some reading. Okay, so this course. So again, as I mentioned many times, no prior coding experience or bioinformatics experience is required. Just hard work is required and a lot of practice. And we'll do Python. I mean, we start with using, then we'll transition to Python, and then we'll transition to bioinformatics. And then the goal is to get knowledge and skills in the state-of-the-art computational techniques for biomedical data science, visualization, and bioinformatics. Okay, so let's review the modules just to give you I know you saw that syllabus, just like to emphasize again. So we'll have 6 modules. We'll start with the module 1, which we're doing right now, Introduction to Code-Based Data Science through Unix Shell Scripting. And then module 2, we'll do data with Python and pandas. We'll use exactly, I mean, precisely pandas library to basically process Excel style files, like CSV files, comma delimited files, right? Like tables, essentially. We'll talk about data visualization a lot because I think data visualization is the key. How do you, like, that's the best way to present your data, right? So we'll talk about a lot about code-based visualization, right? Like how we do box plot, heat maps, like different plots in using Python. Then we'll talk about how to do collaboration and reproducible research for the GitHub, which is version control, meaning that you do iteration of your analysis and you would like to share, I mean, to keep versions, right? So why it's important? Because you can really explore your analysis. You can go one way and say, you know what, that was the wrong way. You can in any moment in your time, come back to any point of your analysis and try different way, right? So all the versions. So instead of creating files like V1, V2, V100, V1000, which is not a good way, you can do version control. And we'll talk about what's that, but that's essential. Then we will talk, module 5 and module 6 will be about bioinformatics. So now you have Unix skills, right, at that point, and sorry, you have Python skills, so now you're kind of ready for bioinformatics. And we'll give you two flavor. I picked up two problems in bioinformatics, which are very common, but also different. So you can get a good flavor of bioinformatics. So one is RNA-seq, so data from human transcriptome, and essentially you want to quantify the genes, right? You want to quantify the gene expressions. So we'll use R code for that because software to process it is in R. I mean, we actually will use both bioinformatics. You will use bioinformatics to calculate. You will use the tool Selman, I think, to calculate the gene expression. But after you get the gene expression, which is essentially the table, you will use some R code to do that, to run some statistical analysis. And module 6, like classical bioinformatics problem of alignment. And in general, in JS, it's next generation sequencing analysis, right? Given the reads from the machines, you would like to place those reads on the human reference to reconstruct your personal genome, right? It's the very, very first step of many, many different type of bioinformatics analysis. Okay, so that's kind of the high level overview, right, for the January. Any questions? Okay, so let's get started. So what is the learning objective, right? So by the end of this module, you should be able to understand Linux, like in general, and appreciate the difference between command line interface, where basically you just type things, you don't have mouse, versus the graphical interface, which you are using right now as you operate your Windows or other machine, right? You can click things, move files around using the mouse. So in the command line interface, you will not be able to do that. So all you have is the commands. If you want to move the file, you will tell the computer to do that by command, right? By typing something rather than moving the mouse around. And then a bunch of commands, which are very, very simple, like very like base level, but I think it's essential to know them. Okay. Following task you will be able to performance using: create a txt file, reorder the file, see what is inside the directory, move the file, delete the files, use grep command to search the text on the txt files. Like, remember I told you about this project where we classify the papers into primary analysis versus secondary analysis. So actually we have used simple grep for that. So something that's quite useful. Use awk, process delimited files. It's a little bit like, how should I say, complementary or similar to the Python. Like, you can do things like that in Python, but sometimes when you use Unix, I think one-line solutions, like you just want to type things as the command rather than write a Python script. I think sometimes it also makes sense. And then you want to use seed to edit the files. If you have very large files, you can actually edit them directly on the cluster. And I think that's it. So very, very basic command for today and tomorrow. Okay. So what is Unix? So Unix-like system, we call this Unix-like because Unix is like umbrella term for like Linux, Ubuntu, and other types. I just prefer to call Unix, like, not to kind of differentiate between them. That's like command line systems which lacks the graphical interface and require inputting code. Like this is what all you need to know about Unix, kind of. And an environment of Unix, often referred as the command line interface, it's opposed to the graphical interface, which is like, where you click mouse basically and you see things as the graphics. So again, in command line interface, you will only see one command line, like a command line where you will type things, and that's it, right? And you will see the output on your screen, like something like this, like here on the picture. So yeah, so like again, that might be very confusing in the beginning. Be prepared. Quite confusing if you didn't use it before, but again, we have time and we have support, so we'll get through it. Okay, so Unix shell. So basically, Unix is listening to you. You can type something, press return or enter, and the command will be executed, right? That's how you're going to communicate with Unix. So again, in normal settings, you would run Unix on the cluster, but again, that's a little bit challenging logistically to get access to everybody in the class to the cluster. So instead, we'll use Google Collab for that. So that's kind of a little bit like a proxy of the cluster. Like you will. I think the essential is that you will have a chance to practice all the commands in the Google Collab in the same way across all, despite the differences in your laptops and the settings. Okay, so in general, for this class, we'll be using two kind of tools for data analysis. One is Jupyter Notebook. So again, the notebook, it's a kind of very big milestone in data science community. It's developed by many people, like as the community, and it's used in academia and industry settings. So essentially what is that is a combination. So you write a piece of code and you click execute. You have cells, and there are two types of cells. Type number one, it's actually probably three types. So type number one is the code. Type number two, kind of the output of the cells, which can be either like a table or visualization item or like some output, right? So you have combination of both code and the results simultaneously in the same structure. So to me, it's quite useful and nice and convenient, and it's confirmed by high number of people utilizing the tool. So we're going to use that, and it's actually right away available in the Google Collab by default. And then Google Collab. So Google launched this free cloud-based Jupyter Notebook environment, which basically executes the code on the Google Cloud server. So we're not going to use your laptop at all. So that's why I think when we put the requirements for this class, we didn't put any requirements about your laptop capabilities. Rather, you just need to have a Chrome. I think Chrome is preferred. At least I know it's working. Chrome and Gmail account. And that's it, like two things. And you're good to go. And then you're using essentially the power of the Google hardware, getting access to that. Again, as the free user, you'll have limited resources, but that should be enough for this class.
So sorry to interrupt. So basically, we can use an Android device, a tablet, an iPad, and whatever is connected to.
Yeah, I think so. I know laptops are working if you have normal browser, but I imagine I never tried actually. So to use Android device or tablet or anything like that, but yeah, I would say probably. But any laptop.
If you talk about Chrome, it should work on all devices because the code is running in cloud, so
yeah,
it should not be a problem here.
Okay, great. Yeah. So if it works, then just like let me know. I think it's kind of, probably it is, but that's cool. Yeah, exactly. Great. So it's really scalable. It's like it can be like run in any device. Yeah, that's good. Okay, so what is Notebook again? So Notebook is an interactive, editable document defined by code. It's a computer program, but one that's designed to be easier to read and write by humans, right? So this is the definition. And again, that's a combination of the code and results at the same time. So why use interactive notebooks? Several reasons. It's a good tool to explore data and look to do exploratory data analysis and visualization. Provides good interface to iteratively, like through iterations, running code, exploring results and visualizing data. You can run code, explore the output, tweak the code, and reevaluate the results, right? That's essential. So you have infrastructure, you click the button and you rerun all the steps. Now you, let's say, go in the middle and say, hey, I want to tweak this parameter and change it. So you tweak the parameter, go back to the top, click the button, and you will rerun all the results, and you will regenerate all the figures, all the tables, and so on. So you can see the impact of tweaking that parameter on the entire analysis. I think that's quite nice, give you a lot of power to explore. And I think that's the key to data analysis, really be able to explore different directions, different settings, different thresholds, and so on. And I think last but not the least, allow effective sharing of visualization and the code with the community as you publish your paper or like your project, or like make it available globally. Or if you share with your collaborators, it's a very effective way to share and sit together and work, or like even remote setting. I mean, like online settings, like share that, and then they can work on it and so on. So that's quite effective. And we use a lot in my lab. So I'm biased here, but I think it's a great tool. Okay, this is how it looks like, right? And this is what something similar, maybe a little bit less simple, but you will be capable of writing as you finish this course. So here you can see we write some code and we write some code to process the tables essentially. And the table is about recall and read length of technology and so on. Doesn't matter, but we just print the table just to make sure like what we do is looking as expected, right? That's a very essential question. What I'm doing, does it look right? Like the output I'm getting, is it like what I need? So I think you need to ask that question at every iteration, right? You did something, like why not print that table and see, oh, that looks good, that's correct. Then you change some colors, you define some palette, and you draw the plots. And those are the plots, right? So again, a little bit simple code, but I think close to that. We'll learn how to write. So for example, and you say here, like, I really don't like the colors, I would like to redefine them. You just go here and change the colors. Or like you say, you know what, in this case, you have 4 colors because you have 4 different tools. But you say, hey, I'm not really interested in tool number 4. Let me exclude it. You can change it here and just click one button and you run all the analysis. So it gives you a lot of flexibility. So this is what we would like to get, right? So this is what we would like to learn how to do. Okay, so where can you run your analysis? You can run on your laptop. Actually, you can actually run Jupyter Notebook on your laptop. I know it's very, very easy to run on the Mac because you have kind of Unix on the Mac. Underneath the Mac, you have terminal. This is what I do. It's relatively easy to run for the newer Windows machine where you have Unix, what you call it, simulator, emulator, whatever they call it. But I know it's a little bit tough for the older machines. You need to install virtual and so on. So you can do that, like if you're interested, and or you can run it on high performance computing as well. So here, again, just we didn't want to waste time, and sometimes it's actually hard to deal with your particular settings on your machines, like of your laptops. We said, let's define some uniform environment will be exactly the same for everybody. And I think it's a good for teaching and acquiring knowledge, because what TAs will be showing you will be exactly the same on your machine, right? So that can facilitate good learning. So we decided to be on the cloud, and luckily Google provides free resources for that. So we're going to utilize that. Even though, like, I think in real life, for the Jupyter, for this analysis of the simple files, we typically use just laptops. And actually for the analysis of the big files, we typically use high performance cluster because it's free. And the reason we don't use cloud for the big files because it costs money, and actually high performance cluster are offered for free for all the students from my university, at least. So we use that free resources, but there is different opportunities. And okay, so you can ask question again, why? So like this is what I just said, that in certain cases installing Unix or Ubuntu or Linux on Windows might be challenging, so we wanted to avoid that. And then in general, I think cloud is an alternative, allowing to run Unix and the Python on any computer, regardless of operating system. And this is what we really wanted. We want to have uniform experience across everybody in this class. Okay, Google Collab. Actually, all the files will be stored on the Google Drive. And you can share it just like any file. You can share it, download it, and so on from Google Drive. And as with any notebook, the file will be like the notebook will have two components: the text block, where you basically write some text, like notes, like I am running this analysis, like comment, and then you will have the code block, right, where you write formally the correct code, and then you will have output. So that's kind of like the structure, but you will see that as you practice. Okay, so again, I will go through the slides. I will not do any kind of hands-on yet. Why is this moving? So, but then as TAs will join in like 20 minutes, like they will work with you to set up Google Collab and so on. So Google Drive, so we'll use Google Drive to store all the files, and they support Google Collab because it's Google basically. And use your personal Gmail address, even though if you have, and probably not the case here, but if you have Gmail from university, sometimes they restricted that, but normal, typical Gmail free email address is enough account to get access to Google Collab. Okay, so how to do it? You will open Google Gmail account. If you don't have one, if you do have one, then go to your Google Drive. And then you will install Google Collab. So you will find the new, more, and so on. But sometimes you don't even need to install it. If it's already available to you, you would not need to install it. So again, the TAs will go through these steps with TAs basically to enable the Google Collab for you. But you can see it here. It's like more app and G Suite, and then here. So this is the sign among Google products you need to find. So it's called Collaboratory. And this is the sign, right? So you need to install if it's not installed, but I think for the Gmail address is already installed. So you can just go ahead and use it. So this is how you would install it. This is, yes, how it will look like. Okay, so one thing will store everything in one directory, which will be on the Google Drive. So you will go data science, data_science_workshop, and then all the files will be there. So basically you will need to connect, which I will show in a second, notebook and your Google Drive. So all the files from Google Collab, you can write and you can read and write from Google Drive. You cannot write to your laptop, right? That's like disadvantage, but you can work with the Google Drive and later download the files if necessary from Google Drive. Okay, create a new Google Collab notebook. So we'll go to new, more, Google Collaboratory, right? And then we will open the new one. So this is how it will look like on the right. So what we'll do, so before that, the best practice is to keep all data sets and notebooks related to a certain project, in this case, let's say the class, into one directory. Let's say to be consistent, and actually will be a great help for the TAs as they troubleshoot like the issues you're having. So it's a great use if you can keep exactly this name. If you say data_science_workshop, let's keep this name. Like, so you want to create this directory in your Google Drive and like inside like your Google Drive, not any subdirectory, but like in the Google Drive directory. And this is where all the files related to this workshop will be. So it's easier for us to help you, but also easier for you to locate the files when you need them. Okay, so as you see this Google Collab, and again, TAs will walk you through, but on the high level, you have two buttons, right? You can add code and you can add text. Again, text has no much meaning. You just say what, like some comment, just to structure your code. I think the structuring the code is the key in the sense that you can explain what you're doing. And I think you should, especially if you're just beginning, right? So it's hard for you to revisit the code later. So adding more comments will help. Then I think if somebody has some noise, if you guys can mute, unless you're speaking, if you can mute, this will be helpful. So then the code, you click the button code and you will create. So you can see here when I click the text and I wrote, this is my first notebook, right? I just wrote some comment. And then when I click code, I got this cell. So then I write the code, then I click this button to execute that, right? So the code will run. And then if you did everything correct, it will run. If you did the type, it will show you, or if you did the bug or error, it will show you the warning, right? Okay. And actually the text is the markdown language. So, I mean, I will not go much into details, but like there is some very basic syntax. For example, if you want to do header, basically if you want to do large letters, you need to put 3 asterisks, I think, or 2. Hey guys, I think there is some noise. So if you guys can mute. Let's see. Mute. Cool. So, and then, yeah, so like 2 asterisks means header 1, 3 asterisks, like stars, means title, I think, and so on. So, but you can also use normal. I think it's also available, the menu for you. So you can use bold, like all this, like normal editing of the text. And then on the right, you will have a preview, right? So what it will look like, right? But it's like just to write you the command to better structure your code, right? So for example, if you're writing about code about something, you want to say, this code is about this, this, this. So later when you visit or if you share with somebody, somebody can understand what did you do. So it's a good practice. Okay, so here, we will not yet doing Python, even though notebooks are designed for Python, but luckily for us, notebooks can also do Bash, which is the language of Unix command line. And actually it's kind of also can do Unix kind of commands, but also can do R. So basically, theoretically, you can write the notebooks, which will have 3 components like Unix, Python, and R all together. Seems like it's working fine, right? Combining the languages, which I think it's a powerful thing, right? Sometimes it's easier to use one language than others and so on. So, okay, how we write the code? So we click again, plus, and we add the code, and we need to say bash, right? So we need to say that we're using bash. So we need exactly this command, 2 ampersands, and then, like 2 percentages, like exactly this line, and then you write your command. So in this case, we'll write pwd. So pwd means, let's see if I have the slides explaining that. I mean, we'll get to that. So pwd will tell you the current directory, right? Where you are. Like, are you in your desktop? Are you like somewhere? So by default, it's a default kind of like directory called content. So not that meaningful yet, but as we connect to the Google Drive, that will be more meaningful. Okay, so to execute, right, you just click this button here, and then you will execute the code. So in this case, this command is supposed to display something, right? It's supposed to tell you where you are. It will not display anything. Okay, admitted somebody. Okay, great. So, oops, sorry, I'm doing this. Wait, I cannot, okay. Sorry. So yeah, so some commands will do something, but will not display anything at all, right? So in that case,
well, I'm just gonna kind of start from again, just going top down, starting with account management, and then going down to the checkout flow, just to address just clarify some questions I have regarding the comments. Does that work for you? Yeah, that works for me.
Okay.
Okay.
So there is some noise from people.
I left a lot of comments, so as you guys probably saw. Is that helpful for you guys?
I think.
I think so.
Okay. Okay, I think it's good. No. Okay, yeah, great. Okay, cool. Oops. So go back. Okay, so yeah, so some commands, right? So sometimes the commands will basically not display anything, right? Sometimes. So you do something, nothing to display. That's fine. And so basically, if you run something, unless there is errors, then you need to be concerned, obviously, and you need to change something and address it. But sometimes the commands will give you nothing. So either nothing is fine, or some output, expected output, right? So this is what the command is. Okay, great. So you're ready to run, like you have the setup at this point to run the Unix command, right? So pwd. So what it will do, it will display the current directory. So you just do pwd and you see the current directory you're currently in. Directory means the folder, like, you know, like other words, in other words. So what is typically the Unix system, right? So like that's typically the files we are observing, you know, like, and this is actually comes from the high performance cluster, but it's something related to your Google Drive, right, as well. So like on the cluster, actually, there is a lot of people on the cluster. So that's why people need to share and don't delete each other files and so on. So that's why there is a home directory for each member. And then you have access only to your directory rather than for everybody else. But on the Google Drive, it's only you. So you don't have that kind of issue. Okay. So basically, when you open the Google Collaboratory, you always start with your content directory. That's just by default how it is implemented by Google. So you can create subdirectories in that directory or other directories to store specific projects or group information. So if you don't connect to the Google Drive, everything is temporal. So and then will be deleted after you end the session. So it's very we highly recommended to connect to Google Drive so you can really store the files in your personal Google Drive. Okay, so another command, we can create a directory, right? Like normally in your Windows, you would right click button, something say create new directory, something like this, right? Here we don't have mouse, right? Remember? So we need to know the command how to create a directory. And that command is mkdir. Actually, it stands for make a directory. So it's easy to remember. Then actually running that command will not give you any output, right? So that's kind of by design, right? If there is no errors, means things are good. Directory was successfully created. So this is how you need to treat that command. And then, but actually you will also see the directory as you create one. And then when you will connect to the Google Drive, you will actually see the directory in your Google Drive. Okay, so then how would you navigate, right? Again, you use the mouse typically in the Windows, but here you don't. So what you do cd, and cd stands for change the directory, right? So you can change the current directory. In other words, you can go to certain directory and check what is inside, for example, right? So here we decided, okay, we created a new directory called test. Why don't we go there to that directory and see what is inside? So actually we'll not check yet. We don't know yet how to check what is inside the directory, but rather we know how to list the current directory. So what means we successfully changed the current directory? Or other words, we went to some directory. What that means? Means that your current directory have changed, right? Where you are currently have changed, right? That's your expectation. So I think you want to go by kind of common logic, right? So when you run the commands, especially in the beginning, try to check yourself every time you run the command. Did things went as expected? And here, the things went well or the things went as expected will mean that you have changed your current directory, right? So if you say cd, change a directory, new directory, which is just created test, and then you say pwd, you type those two commands, you should see output. So you should be in that directory, right? Okay, so we know two commands. Okay, that's a good start. So in other words, if you think about the tree kind of thing, right? So then here you have content directory, and then inside the content, there is a test directory. So one operation is to go one level up, right? So if you are in the test, you can go one level up. And there is only, if you think about the tree structure, there is a unique way to go up. So you don't need to say where, because there is one way to go up, right? Think about the tree. If you draw the tree, computational, like the tree, you cannot, let me show you again here, right? If you go up on the tree from bottom to the top, there is one way to go up, right? There is no ambiguity. If you go down, sure, there's multiple ways. That's why when you say where to go, you need to say exactly the directory. But if you go up, you just say up, one level, all you need to say, right? Okay, then if you don't like the tree, that's fine. Like you can think about the absolute path. And the absolute path is like this, content test. So meaning go one level up will mean to go from the right to the left. So if you're in the test directory, one level up will mean to jump one level from the right to the left, and you will end up here. So anyhow you would like to visualize that, but there are two ways to kind of visualize that. And then there is ampersand, meaning go back to your home directory. In your case, home directory will be directory by directory content, just because it was implemented like this by Google. But if you're using some other settings, home directory will mean something else. All right. So we're going to practice. And again, we will do this with TAs. So you will do CD ampersand. We'll go back to your home. I think it says root here because probably the screenshots are not from Google Collab or like something else, but you will figure out that. Then you can go one level up and you can do CD. And CD will actually the same as CD ampersand. Ampersand is this sign, right? So CD ampersand here. Oh, sorry, tilde. I'm sorry, I forgot the name. So CD tilde, it's not ampersand, tilde. So CD tilde is exactly the same way as CD, right? There is no difference. So this sign is tilde, I apologize. It's not ampersand. Okay, so we know CD tilde is the same as CD, go back home. And CD two dots, like this, is to go one level up, right? Okay, so we more or less know how to navigate. We can go down, and we can kind of go back home, or we can go up, right? So we got something. Okay, so another thing is, if you would like to create some directory with the very long name, you can do that. So what's the point? Then if you need to go there, it's quite inconvenient to type all these random letters, right? But if you don't type them correctly, you will make a typo and you will fail to go to that directory. I think that's one challenge. If you, with the command line, you will observe even the tiny typos will result in errors, right? If you forgot to put space, you forgot to do something, like you slightly change the syntax, things will fail. So that is a challenge. And to address the challenge, it's just practicing commands. So, but don't be discouraged. Often you need to repeat a couple of times and look at that command for a good number of minutes until you notice the type. And especially if that's new for you. I mean, for me, probably I look a second and I see because I saw those commands for many years. But since you're seeing them for the first time, you need to spend some time looking at them to find the typos, if that's the case. So here the point is sometimes you can make your life a little bit easier by using some smart decisions. So smart decision in this case to use autocomplete. Actually, you can just type the first letter of that directory and press the tab, and you can autocomplete. So again, with TAs, they can show you that. Okay, then another good command is to type to see what is inside, right? Remember, we created the directory. We want to know what is inside. Or we just go to some directory. We want to know what is inside of the directory. So in that case, you can see that the command is ls, list. It's basically list what is inside, ls. If you have many files in the directory, you will see them. In this case, let's say in this particular directory, we have a lot of things. So they will list them all. It will list all the directories and files inside the current directory. And this is what another output in your notebook, right? It's not only the tables as we'll have the Python or figures as we'll have with the Python, but rather here, just like plain text output. Okay, so then actually when you do ls, you can be a little bit smart and display, especially if you have many files, you can display them as kind of a nice way. You can say -l, which will be long list. You can say minus T, you can sort by modification time, minus S, capital sort by size, minus H, actually to present the size of the files in the human readable format, and minus R, which is reverse order, right? So like, for example, you sort by size, but you want to reverse, you just add more R. So you can combine those, right? For example, we say minus LH. You can see what is L, long list, what is H, list files in human format. So you will get a good kind of table with the files, but then the size will be human readable. What that means? For example, if the files is 3 gigabytes, it will not tell you in bytes because it's inconvenient. There's a lot of digits you need to count and say, oh, is it like, how much is that? But it will really tell you 3 gigabytes. For example, like this, right? So if you have here for 4,000, around 4,000 bytes, it will tell you kilobytes, right? If you do human format, if you say ls minus L versus ls minus LH. Okay, cool. Okay, so this is how we'll download the files. So then you need to type exactly those 2 lines. So that's you will mount the Google Drive, right? In other words, you will connect Google Drive and you Google Collab, so you can write and read the files from Google Drive. So it's a little bit like tricky in some sense. Again, practice is the key, but that's, I think, one of the most tricky somehow things. So it needs a couple of steps. So basically you need to type those 2 lines exactly like this, press here, right? Remember to execute the command. And after that, you will need to go to this URL in your browser and you need to get the code. And then you need to get the code and you will type like the code you were provided with. Okay, so we have, okay, so maybe a couple of more minutes and then I will introduce you to TAs. Okay, so then you will have another thing to allow. So basically it's a couple of steps you need to allow. You need to connect your Google Drive and Google Collab. Okay. And then you need to copy the code and you will insert the code here, right? And then things will connect. How you know that? If you say ls, now you don't see that content. Remember that directory content? You will not see that anymore. So instead you will see Google Drive and My Drive, right? You can do ls, and actually you can see what do you have in your Google Drive, right? Here through command line for Unix. All right. So if you want to know more how you do that, you do it either through manual or you do Google, right? So like I think manual is the old-fashioned way. I think Google is more dynamic, gives you kind of more concise and more kind of personalized information, if we will, because you're asking a particular question in Google, rather than manual, you need to read the entire thing. So, but I think both are valid. You can use both. Okay, so if you want manual, you say mn, ma, like manual, man, ls, and you can see what is there. If you do Google, you can actually say ls sort by date. Actually, I did a typo here. Maybe you noticed that, right? Not data, but date. But even then it was fine, right? So Google is quite robust. So then you see the commands. And now I think you can make sense of them, right? You heard ls, right? Remember ls? So you ask, how do I sort by date, right? I want to see the files by date. And actually there is a quite decent answer here. I think in the first link you click, you see ls minus t. It actually reminds you ls minus l we have using. So why not minus t? What about tr? That should make sense. You know already R is reverse, right? So you combine T and R. So it's not rocket science, just practice, right? But you can ask Google. You don't. So the point is, let's say you know ls, but you forgot all the commands. That's fine. Just ask Google. And then as soon as you understand what is ls, you can make sense of it, right? So I think that's the model I will encourage here. Get some basic knowledge in the class, but then you will be effectively using other resources to educate yourself further. Okay, I think a few more slides and then so I will be done with the lecture part. So we'll use asterisk or like star, so which will be the wildcard, meaning that, for example, if you have 1,000 files in your directory, not that convenient to find all txt, and you really need to find all txt files. Not that convenient, right? It's a little bit like challenging. So what you do, so you do ls ampersand, ls asterisk txt, and you will see all the files and directories which are here, right? In this case, we only have one, let's say. So you can actually not just say what is in the end of the file, but you can say you can say, I want to see files which start with T and ends with G. You can say that. So basically you say T asterisk star and the G, and you can see that. And the same, you can say, I want all the files which start with T, right? Okay. Okay, I think I will stop here. So you will have enough time today. I think the goal for today will be for you with TA to work with TAs, which I will introduce you in a second, and learn how to connect Google. So I think the minimum for today for you to be able to enable Google Collab, connect it to Google Drive, run a couple of commands, and basically familiarize yourself. You should feel comfortable running those couple of commands. So that should make sense to you. And I think for tomorrow, if you have time, revisit the slides, read the slides. Like if you have questions, please do ask tomorrow. I will, when we talk, we start the lecture. And then you will have a kind of a more formal homework, which will be provided as we finish the module 1, which will be essentially repeating all the commands and rerunning them by yourself. So they should work. For example, if you don't mount the Google Drive in the correct way, nothing will work, right? So you want to take time to accomplish that, but it will kind of provide you like the good indicator that you are doing things correctly. I think self-assessment in some sense is very important. All right, so I will finish here. So let me stop sharing. Then I will introduce you to the TAs, unless there is questions. If you have questions, that's a good time to ask me. So, okay, any questions? Okay, so I want to introduce you to TAs. So I think I see Cynthia here. Hi, Cynthia. So Cynthia is a researcher in our lab. She's working with immunogenomics and TCR receptors and BCR receptor for vertebrate species. So she's very knowledgeable in bioinformatics and data science in general. So you guys in the good hands. And she will walk you through from the beginning of Unix. Skipping kind of theoretical part, but like kind of jumping on and kind of ensuring, helping you, each of you, so you can like install, not actually install, but like utilize Google Collab. So I think you have 1 hour for that. So that should be enough. I didn't cover much, actually. Cynthia, I didn't cover, as you can see, I didn't cover much of Unix, which probably will cover more tomorrow. Today, we kind of talk in general about things. So, okay, so then we also have Aditya. Aditya, are you here?
Yeah, I am here.
Okay, great, Aditya. So Aditya is undergraduate. So he's joining us from India. So I think time difference, it's not that good. It's good. It's better, I think, for California than, unfortunately, at this time than for India. How much is in India now? Is it night? Is it day? Is it?
Yeah, it's like 1:24 a.m. right now.
Oh, wow. Yeah, thanks, Aditya. Thanks for your commitment. So it's an even worse time, sorry about that, like than in Moldova. Cool. So Aditya is undergraduate in India, which very strong plans to do research and then apply for the top schools in US for the computer science. So he's coming more from computer science background, but he's doing great in bioinformatics and already preparing his first publication to be submitted. So he's very knowledgeable in computer science and obviously Google Collab and bioinformatics. So he will be together with Cynthia to help you around. So I think, as we discussed, Cynthia and Aditya, right? So we'll not do breakout rooms, right? I think this is what we decided. So instead, if somebody has a question, you can walk through or work through that question kind of in front of everybody so that everybody can learn from each other's mistakes. And I think I would like to emphasize that again, if I didn't do enough, I think it's expected for you guys to be confused, okay? So that's expected. I don't expect you to like open things and say, okay, it works. No, that will not be the case. So things will not work. So, and then TAs are here to help you. So if something doesn't work, raise your hand or like whatever, speak up and say, doesn't work, share your screen and you can work through. And perhaps that will be extremely helpful for others who potentially and mostly probably like struggle with the same issues. All right. So anything else? Any questions for me? Because I will mute myself and stop the video, and then I will. I mean, Cynthia and Aditya will take care of the rest. All right. So actually, Cynthia, I think we'll figure out maybe later today in terms of co-hosting. I didn't have time to do that. Okay, let me make you co-host now, right? I think I can. Stop video, make a host. We don't make you. I mean, actually, Cynthia, what do you think? Actually, I found that you see that I'm recording it, right? So there is a record now going. I mean, we'll edit the video, obviously. We'll remove all kind of conversations. But what do you think? Should I make you co-host, perhaps, right? We'll give you enough privileges?
Perhaps. I'm not quite sure.
Because I'm worried if I do you host, will I? Because I'm recording, which is very important. I want to share both the slides and the video later today. Like if I make you the host, I'm worried that the recording, because the recordings goes into the cloud now. So I'll access the file later. So I wonder, so probably co-host, you will, as the co-host, you should be able to share the screen. I think that's all you need, right?
Yes.
Okay, cool. Let me make you co-host. And maybe Aditya as well. Make co-host. Let's see, Aditya, where you are. Okay, I see you here. Okay, make co-host. Okay, guys, you both should be co-hosts. So you should be sharing the screen and so on. And probably enable others to share the screen. With Notes works, people can send you screenshots and so on. So let's see. If some issues, we'll address them tomorrow. Let me know. So then, if no questions for me, I'm muting myself and stopping the video. We'll be around. Okay. I mean, mute, not unmute. All right, so let's get started. So the plan is today to finish the Unix part, right? And get your basic commands to navigate the system and open the files and so on. So I would say that's like alternative way to like, I think you can kind of survive not knowing Unix, but again, if you do anything related to bioinformatics, I think that's essential. Okay, so we'd like to see how we display the files, right? In normal kind of settings, if you have a laptop, you would like click the button and open the file, right? However, guys, I think there is some noise. If you don't mind, can you mute yourself? Can everybody mute themselves? So I think it's like there is a various way to display the files in Unix. And again, in normal settings, you click the button and you open the file, right? Like in like you open Excel or whatever the file, you have TXT file and so on, right? You use the mouse. Here we don't have the mouse, right? So we need to know the command. So there is a special command to open the files, and there are actually a couple of them, and we'll discuss all of them. So there is a command cat, like cat, then less, and there is head and tail. So 4 commands, and we'll talk about each of them. So command cat is very simple one. It just displays the entire file. So now if you will open now your notebook, right, and you will say cat test.txt or any file, then you should get this kind of message, or whatever the message is in the file, right? You will see the content of the file on the screen. And then okay, so okay, so I was admitting I need to find a way. So now we need to, I mean, that's fine, but imagine the file is large, right? The file is like 10 million lines. Do you really want to see all 10 million lines? Probably not, right? So then that command will be not the best one. So what else? Like we don't have scrolling, right, per se. We cannot scroll. But there is a command which kind of will scroll the file, right? Meaning it will, I think there is some noise, guys. Can you mute? Maybe that somebody new came. So like what means scrolling, right? If the file is 10 million lines, you cannot see it all, right? There is no such a screen which will display you 10 million lines. So what is the alternative? The alternative is to see the file through the lens of your screen, meaning that you will be only able to see the portion of the file, right? So imagine you have a huge, I mean, like a huge file. So you're looking at that file like fractionally, right? Like whatever the screen allows you. And this is less, right? So when you do less, you will see the file fraction of the file which fits the screen, and that's it. So for example, you have a file of 10 million lines, you will only see 50 lines, let's say, whatever fits your screen, no more. Now, how do you change that? You can go up and down like the normal keys, and you will shift that window. So you will see not first 50 lines, but let's say from line number 2 to 51 and so on, right? So you can like change it. Now, if you want to scroll faster, kind of you want to be here and then like move it to the size of the window, then you do space and B. And you can see they're conveniently located above each other. So I think that's the reason they choose those two keys. So you can scroll faster. So it's extremely convenient for the extremely large file when you're in the Unix, right? Like in the system, in the command line. So if you want to, if you're done looking at the file, you will say Q. You will press Q and you will exit. All right. So we will try that. So you will try to open some large files with the TA. I mean, relatively large. We will not give you yet large files. And then you will try to go back, I mean, up and down and see how that works. Okay, there is another two nice commands. Again, very good one for the large files, and they are so they are head and tail. So head will display 10 first lines by default of the file, and tail will display last 10 lines of the file. And if you don't like the number 10, you can change that. You can say -n, and you can say whatever the number. Let's say you want to see 3 first line of the file or 3 last lines of the file. Sure, you can. So this is the tail. So this is how it works. We have a file here, right? If we do head of the file, we'll just see 10 lines, as you can see here, 10 first lines. If you do tail, you will see the last 10 lines of the file. If you do tail -n3, meaning that you say, I really want to see only 3 lines of the file, fine, you will see only the last 3 lines. So I think that's pretty much how you display the files, right? So you can see you have a variety of things. You can just like dump the entire file on the screen, not good idea for the large files, and kind of other alternatives. Either you scroll, basically, or you display a fraction of the file. So whatever whatever the way works better for you. I mean, personally, when I operate Unix, I think I use both. I mean, I rarely use cat because I mean, like there is no much point in that, but I usually use head and tail just to see like beginning of the file and I mean, the end of the file and so on, and also less occasionally. So I think those are good options which you can use for real data. I think the idea is that you only, when you work with the large data sets, you will, even though you will not process the data sets per se manually, but it's always a good idea to look at the data. And I think my philosophy is like always stay connected with the raw data, despite you will run some pipelines and they will do the job for you. But look yourself, just like spend a couple of minutes and look at the data and make sense and make sure it looks like as expected. You can expect crazy things from the data. Somebody forgot to do something happened, and so on. So the same you will see comes for the sequencing data. Like it's often the experiment fails and so on. So it's like things can go off. So you really want to stay attached and as close as you can to the raw data. And Unix gives you that opportunity. Okay, so now let's talk about how we copy the files. So I just want to emphasize there is 2 different ways kind of you copy the file, and they're fundamentally different, right? So you can copy the files or you can move the files. So, and then they are very, very different. So when you copy the file, you're creating the copy of the file, right? You're like looking at the file and creating identical copy somewhere else in the system. So the reason I put time here, because it's time consuming, meaning the computer needs to go through each line of your file, right? And copy it somewhere. So it's time. It's like, it might take a lot of time. Also space. Basically, you're doubling the space you're utilizing to store your data because you have 2 copies now. So be mindful, like only do that if you need it, right? If you really need 2 versions of the file. So what can be the reason for it? The reason can be that you really would like to modify your file. However, you want to preserve the original one. Sure, that's a valid reason. But if you just say, I want to run 2 types of analysis, but I will not change the original data, that's a bad idea. That's not a good one, right? Because you will not change the original file. You use the same one. Or let's say somebody shares the file with you, right? Some collaborators and so, and you say, I would still need to change it, right? I need to change that file, but I really want to preserve the file somebody else gave it to me. Sure, you need to copy it, not to affect the original file. Now, what is move? So move is just like physically, not physically, but like moving the file to the different directories, right? So you don't create any new copy, you just move the file around. So that's good command to organize your files. So when you do the projects, I think Cynthia mentioned that a little bit about creating multiple directories, being very structured. And I think as the project gets complexity, it's essential and absolutely crucial to organize your data kind of mindfully. And I did personally run so many times into trouble of not doing so, saying, oh, I will save time. It's a simple project. Let me just dump everything in one directory. Not a good idea. So and the same with the file names. We'll get to that, but I think try to call your file names the real names. Don't say t1.txt. You will not remember, at least I cannot remember what is that, and I'm so confused later. So spend time, organize your files, like have the file, like have a directory called, for example, like as I think Cynthia said, like raw data, have a directory called summary data, and so on. So that will help you. So move is the command to help you with that, right? Like move files around. Now, I put the sign, you can see somebody's running very fast, meaning this command is very fast. Why? Because on the hardware level, you're doing nothing, right? You have a file stored somewhere on the hard drive, somewhere. Now you say, I want to move it. Computer will not do anything. Computer is smart in that sense. It doesn't need to move physically. Instead, it will change the label. Say the file is kind of connected, is located to this directory of that, but on the hardware level, it will stay exactly in the same location, right? Meaning it's fast, it's easy to change one string. So that will be almost instant, and copy will be long. Okay, so I hope that's clear. So I hope that's clear what's the fundamental difference between those two things. And now remove, I think remove is simple, you just remove the file. And I think it really depends on your system. Do they have a backup? Sometimes they have a backup. Some clusters are tough and they will not have a backup. So if you remove something, that's it, it's gone. So I think it's kind of stressful. So we'll talk a little bit about remove, and I will tell you what is the best way to avoid the situation of deleting, like what can be done to avoid possibility of deleting important data. Okay, so any questions so far?
So in case, like, sorry.
Mm-hmm.
So if you remove a file, we don't have like, depending on the operating system, we usually don't have like a recycle bin in Windows or something like that on Mac.
I mean, it depends. Yeah, it depends on the, not in Unix, not in Unix. Yeah, I mean, in Unix, there is no recycle bin. I think there's sometimes backup, but it depends how the cluster or the system or the cloud or whatever it is configured out, right? Like whatever the system admins decided to do. If they decided that things can be restored, it can be restored. But I ran a few times in my life where I was operating the cluster and then basically I deleted some files which were kind of important, and that's it. They couldn't restore it for me. So it's kind of a mix. I think you need to just know. I mean, you need to ask people who like running this cluster, or like, like figure it out, like what is the current system. But not kind of, you don't have this bin. I think bin is kind of for more like laptops or like more user-friendly. Unix is not that user-friendly in that sense. It's more rough. But I think there is a reason behind that. You need to be effective. Data is large. If you create recycle bin, I think you will just easily kind of like you will get so much space taken by it.
Yeah, but people make mistakes, so that's.
Yeah, I mean, but we'll talk. We have two options. I will kind of present you two options what can be done on Unix, but not recycle bin. Actually,
sorry.
Git, I suppose.
Yeah, Git, like if you commit, but if the data is large, you cannot do Git. I mean, if you have space externally, but again, if we are talking about large files, it's not always the possibility.
Sorry, I would like to interrupt you a little bit. I created an invite link to Slack. So
Okay.
anyone who wants to join, please follow the link in the comments.
Okay, thanks, Mihai. Can you send to, I don't know if Vlad did already, but can you send to Cynthia that and say that?
Cynthia, I don't have email.
Oh, I didn't
Any email.
see it. Oh, interesting. Okay.
No.
Okay, fine. I think I need to put, okay.
Sergey, I will CC Cynthia right now with Mihai's email. So
Okay,
I
great.
just sent her
Yeah, yeah.
message. We will
Yeah,
figure this out.
Yeah, okay, sounds good. Yeah, thank you. All right. So, okay, so yeah, we'll talk about remove a little bit more, what can be done, but that's kind of fundamentally where we are. Any other questions?
Yes, maybe if possible, a quick question. So we have one command, MV, yes? So, but in fact, we can have different possibilities with this command, either move or rename. Last time we used it to rename a directory, but to use it to move, we need to use in another way, the same command.
Yeah, I mean, I think I will have the slide on that, so I will cover it.
Okay, yeah, thank you.
That was a good question. Any other questions? Yeah, now we will discuss each command kind of in details, right? So CP, copy, right? So to copy a file, use CP, right? Original name, source, right? Original file, source, destination. Basically, source is the original file, destination is the file which will be created and which will be the copy of the source, right? This is what we mean by copy. And then if you copy if you copy the directory, you need to say -r. So the R, if you're curious, the R stands for recursion, I think, because if you're copying the directory, you need to copy all the files inside the directory and directories inside. But how about you have another directories inside those directories and so on? It can go far away. So you kind of need recursively to take all of them. That's why it's -r. Okay, so two commands, right? Like CP and CP -r. Let's see some examples. So if you want to copy CP file name, original file name, test.txt, that's original one, test1.txt, that's a new one, which will be created. So that's the command you run. And then if you check what is in your directory, you should have your original file, which is here, test.txt, right here, and the file which was just created, test1.txt. Okay, so that's how you create a copy of the file. If you want to create a copy of the file and simultaneously and place that copy somewhere else, not in the current directory, so you need to have that directory first. So this is what we do here. We create a new directory and we copy that before that, so we just copy the directory. So hold on that. So here we just copy a directory. We say that's a previous one, new, we just created, and we want to create a copy of that directory. So we say CP -r new, and here new2. So new2 will be created. So the difference, as you can see, the difference between two commands is just -r, right? If you do directory, don't forget -r. If you do files, you don't need -r. Okay, now you can actually have a couple of more possibilities. I mean, they're straightforward, but I want to cover them all. So you can copy the file somewhere else and preserve the name, right? So here we created a copy and we give a different name of that file. But here, when we say cp test.txt new, we actually are copying the file somewhere else and preserving the name. Or other words, we're creating the copy of the file with the same name somewhere else. So I think that's one. And another one, you can copy the file somewhere else and give a new name. And this is what we do here. We do cp, name of the file, and we say where we want that file to be copied. We say, okay, we want that file to be copied to directory new. Good. And do we want to give a new name? Yes, we would like to give a new name, and the name will be test_new.txt. Okay, so then we go to that directory new, and we expect to see two files, test_new.txt, the result of this command, and and then test.txt, the result of kind of this command. Okay, so I think that's all the possibilities you have when you're moving the files around. Not moving, copying the files around. So I think that those kind of more or less cover all the cases. Okay, so that's all you need to know about CP. Now, let's talk about move. So when we move, it's easier. We don't need -r option, right? So when we move the directory or we move the file, we just need to say mv command. And then we say source, the original file, and where do you want that file to be moved, destination. And then, as it was correctly mentioned, it can be also used to rename the files, right? The same command. So let's start with moving the files. So we go to our directory test, and you know the command for that, cd, right? Change the directory, test. Then you say pwd. Pwd means tell me the current directory. Usually when you, my personal choice is like when I navigate the directories, I want to make sure that I went to that directory of my choice. So I do quite often pw just to make sure, oh, I'm here. That makes sense. I want to be here in this directory. Okay, so we will move the file test1.txt to the directory new. Okay, so we say move source destination, right? So what will happen, the file will be moved from the current directory, which is test, and moved to the new, meaning it will disappear, right? It will disappear from the directory test and will appear in directory new, right? This is what we mean by move. Okay, so straightforward. Now, if you want to rename the file, it's even, I think, simpler, just old name, new name. Right, so we say we want to rename the file called test.txt, and we would like to give it new name, and the new name will be test_rename.txt, right? And then we do ls, and we want to see the results. So basically, that file test.txt is no longer here among the file in your current directory because it was renamed, right? So you have test_rename.txt. That's a new file. So you can do two things at the same time. You can move the file and rename it. How about that? So you say, I would like to move the file, which is called test.txt, which is actually should be a different file because this one disappeared already, right? Remember, we rename it. And I would like to move it to the directory new, but also give a new name, right? So you would like to say, I would like to give a new name called test2.txt, right? So you're moving and you are renaming at the same time. Okay, so that's all I think about move we need to know. Any questions so far?
So as understood, we have some commands available on Linux on Collab, right? That's why
we'll try, Cynthia will try to, again, like we're doing this proxy of Linux because we as the class, we don't have access to the cluster. Yeah, so Cynthia will work through those commands, and try to, I think you can use those in Google Collab.
Yeah, I see. We have MV, we have CD, but I tried to do touch, but it's not working. I can't create any file. So it seems that part of them works or part not.
Which one you cannot use?
Touch. Create a file.
I see. What about VI? Probably also you cannot, right?
VI?
Yeah, VI. Also probably not. Yeah, again, there is some limitations. There is some limitation.
It's a short version.
Yeah, not even the short version. It's kind of not real Unix. Like it's some kind of version of common, whatever.
Secure shell.
Yeah. So some commands will not work, but yeah, we'll try to do the commands we can, and the rest I will just tell you how to do that. So when you get access to the cluster, you will know what to do. I think this is how we'll approach this. Okay.
Thanks.
Cool. Yeah, thank you. So another thing is kind of in between creating the copy and then moving the file is creating the link to the file. I think very similar how you do like what is called shortcut, right? How you do the shortcut of the file on your desktop, right? So you have a file which is you often use it, right? But it's located somewhere far away on your laptop, and you say, I want to have it handy on my desktop. So what you do, you create a shortcut to that file, right? And you have a link to it on your desktop. So the same here, we can create the link to the original file, right? So this is how the data is, right? You have a original file which points to some data on the hard drive, right? So that's how it is. Now you say, I would like to create a link which points to the original file. So the only reason to do that, you want to have it handy in some directory. So for example, you have a directory where you store the raw data, but you don't want to move raw data because you say, I want to keep it there, not to mess it up, not to touch it, and so on. Now you say, I want to do the analysis, and you create a new folder or new directory called analysis something. And instead of moving the file to that analysis directory, again, because you want to preserve the raw data, but also you don't want to copy that file because remember, it will double the space, it will take time. But you say, how about I just create the link? So in that new directory called analysis, you will have a file which actually looks like a real file, but in reality, it's just a link to the original file. Okay, so this is the command will allow you to do that. -s, original file, and then the link name. So few kind of important points about the link. You can do anything what you normally do with the file with the link. Like you can actually copy it. You can remove it. You can actually look inside. You can count number of lines of that file, like whatever you think you should do that with that file. But don't change the file, because if you attempt to change the file, then you will affect the original one. So don't modify the original file through the link. This is what you should not do. Okay, so I think we'll have some examples a little bit later about link. Okay, so the last command is the remove, rm, right? So remove and remove -r. If you would like to remove the directory, you should say remove -r. If you would like to remove, you just say rm. Okay. So I mean, again, just like to be on the conservative side, because it's sometimes the possibility that the files might be gone forever, at least on some Unix machines. So be mindful with that. So what we can do, okay, before that, so if you like examples, which you will work with Cynthia, hope. So we will remove the file rm, name of the file, rm -r, name of the directory. Like that's it, right? Like 2 commands. Okay, so let's.
Sure. Sorry to interrupt. And if we don't introduce the this R, and when you remove a directory, will the other subdirectories will be removed? Or
yeah, that's why. Yeah, exactly. That's why you say -r. Everything.
But if you don't put a -r in a directory, will the subdirectories remain?
Probably it will not work. It will not work. No, you cannot delete. It's kind of weird. Like you cannot delete the directory without. I mean, the whole definition of deleting the directory means remove everything inside, right? I think that's the definition.
Throw an error. We'll throw an error. It's not possible.
Yeah, I think it will get the error because the Unix knows that that's a directory rather than the file, so it will tell you this. Like you cannot. It's incorrect that you are removing the directory as the file.
Will work in case if a folder is empty. In case a folder is empty, it will work without R.
Okay, cool.
And I have another question regarding the shortcut, the link one. If we move the original file, will the link be updated somehow, or it will?
No, it will be broken.
Okay.
Yeah. If you change the original file, I imagine link should be broken. Actually, I never tried, I think, or like I tried long ago, but I think so. You can actually all those questions, as soon as you have, so basically that's the rule of Unix. So I think you will get soon enough background so that you can, I mean, sure, ask me, please. I mean, I'm glad to answer, but I think those questions, like you can actually check yourself, like you can create the file, move, like and move and see what happens.
Yeah.
I imagine it should be broken, but we can double check. Oh, ask T. How about that? Ask Cynthia about that to show you that if she can, and she will show you how it will be broken.
Okay.
Great. Yeah, but yeah, I'm glad like you guys asking the questions, and then I'm glad Mihai you are trying things, so you will keep us updating about like like what will happen if. Okay, so so a few more things, like you can copy multiple files at the same time. Great. Or you can remove multiple file at the same time, right? So if you want to copy multiple files at the same time, you will just say all the file names you want to copy and then where, right? Like here, the directory name. And if you want to remove, you just say all the files you would like to remove. You can use asterisk, like the star. So be careful with that again. So you can remove, with move it's fine, right? You can move the files, for example, starting with F, all the files starting with F to some directory, like new in this case. So, and then you're kind of, I hope you're kind of getting the point where we are going with this, right? Like why it's nice, right? To command line. For example, imagine the situation you're dealing with like in graphical interface when you do mouse and so on. So imagine you have a directory with thousands files. It's like a mess of things and so on. And you need to organize it quickly. So you can, I would argue will be hard to do something like this. I mean, sure, you can sort it, select it, but this becomes extremely efficient. As soon as you get some skills, it becomes extremely efficient to organize files and so on, especially large ones, like thousands, 10,000 files, because you can do it quickly because you can move files with the certain properties around, right, and organize it effectively. So it's all about like doing not per file, but like per many files at once. So then about remove, you can remove all the files starting with F. Sure, you can. Then you can remove all the files starting with L, starts with L, ends with S. Please, you can do that. Or you can remove all the files which are which ends with txt, right? You can also do that. Okay. So let's see, do I have an example? Okay, so what will be the most extreme example of remove? You can say remove star. So you will remove all the files in the current directory. Quite dramatic. Then you can say remove -r star. So you will remove all the files and directories in the current directory. So those are sometimes necessary commands because you want to clean the space and so on. But again, be very mindful when you use those, especially in case you don't have like a backup. Okay, so actually, do I have those slides on the backup? Let me see. Okay, maybe not. So I will just then explain. So I think there are two ways you can work with the kind of like preventing loss of the important information. So I think one way will be you can do, which again, we will not be able to do in Google Collab, but it's generally that's possible on the Unix. So you can do alias. So meaning that, I mean, first of all, when you run those commands, you can run them in the, I think it's -i option. I need to double check. So the option basically will give you the prompt, will give you kind of safety message. Look, do you really want to remove that file? Right. So, or for example, if you're trying to move the file and if there is a danger of overwriting some file, right? Because you move the file from here to there and that file already exists there. So you will overwrite it. So by the current setting, it will just be overwritten and you will not get any warning. So if you would like to get the warning, I think you need -i option. Now, then if you're trying to remove the files, you will be at least asked, like, do you really want to remove that file? Kind of similar when like you have it on laptop. You say yes or no. But it's not convenient to say -i option every time because you might forget it. So what you can do so-called alias, and then by the way, this information is like extra, just like for those who you're curious, it's not kind of required for the class. So if you want to do alias, so what you say every time I will run move, in reality, move minus I will be happening, will be running, right? This is what you would like to define. And I think I personally did on my Unix machine like this. So I'm getting this message. Like if you want to remove the files, yes, no. That's one way. And I think another way, just like back it up. I think make a backup of the important information. If the system doesn't do it for you, backups, I think the only the only possibility, you should do it yourself. And then I think you don't even, one possibility to move externally, right, somewhere. But if it's not the possibility, in sense, like if it's too much data or too much time or something, you can also do locally. And actually what you can do, you can change the privilege you have for those files, and you can take the privilege from yourself to modify the files. So to me, I think that should partially solve, right? So because you cannot accidentally remove those files because you don't have rights to do it, because you have taken it from yourself. So I think, to me, that's two ways I'm kind of dealing with accidental loss of the data personally, but maybe there is more advanced way. Okay, but that was extra information for those of you who are interested in this. Okay, so any questions so far? Okay, so that's your summary. I would say, like if you want, you can print it and have it handy. So you remember those. So those commands should be familiar for you right now, right? I think there is a typo. It should be CP minus R and the space. We forgot to put the space. I hope there is no more. So for VI, again, we'll not kind of like, we cannot create the files, I think, on the Collab, so we'll not do that. But I think the rest of the commands should be familiar for you. So, that's the very essential commands. Okay, so any questions so far?
But we can create with touch.
You can on the Google Collab?
Yeah.
Okay, great. So let's do touch. Yeah, we'll be using touch, and Cynthia will show.
Yes, we used touch last time, but last time they also showed CP minus 2 minus R. Yes, not just minus R, but twice minus was.
Why? Why was it twice?
I don't know. I didn't understand, in fact.
Did anybody else understood like why it was minus?
I think it's the same thing now. I googled it.
Why? Why do twice? Like, what's the point?
It's the same thing now. I'm not sure exactly why, but it's the
Okay,
same
I can
thing.
ask Cynthia. Actually, can somebody email to Cynthia and ask this question and CC me? And then we'll figure out that.
Because like when I installed Ubuntu for Windows and Windows 10, you could define like the version, and usually you can find like bash double minus and write version, and it will pop a version, or 1 minus and version. It's the same thing. So I'm not sure exactly what's the difference between these two.
Yeah,
but I suppose
actually I always use minus, like 1 minus. So, but let's ask Cynthia. Yeah.
I have maybe a version. Then sometimes we have two version. With 2 minus we use short version, minus version, and with 1 version minus V. We have two declarations for the same thing. And other thing can be some kind of explanation, maybe some conflict with the internal commands, and to not conflict to other commands, they added 1 more minus.
No, but wait. So when you talk about two minuses, which parameter are you talking about? Minus R or minus V?
Minus R.
I see. Okay, that's
CP minus R. Yeah, maybe if just 1 minus R, 1 subcategory, and if minus R, 2 subcategories. I don't know. Can it be so?
Yeah, I don't know. So maybe maybe Olga, do you want to send email? Maybe you, if you don't mind, send email to Cynthia, ask that question, CC me, and we'll figure out that, because I'm not familiar why there is 2 minuses, but we'll figure out this.
Okay.
Great. So any other questions? Okay, so that's the same commands, right? It's like this version, if you like this version better than the other one, you can use this, but that's essential commands you should know. Okay, so I'm not sure, was Cynthia using those working materials, or like she just gave you the files?
the first lab, we had some explanations, and then the first assignment.
I see. Okay, so maybe I'm not sure if we'll be using that, but if so, she will cover that.
Okay.
So, okay, so I think it's like, so we will skip some because like, I think I really want to give it just essential. I don't want to kind of overwhelm you. So we'll skip certain parts of it, but we'll kind of cover the essentials. So let's see what we would like to cover. So I think very important definition is for you to understand that's a relative versus absolute path, right? So relative path means it's relative, right? So you can imagine the address. If you say the full address, you know, like the country, the state, like in the US, for example, and zip code and so on, that's an absolute, and the country. So that's absolute path. But if you just say, but if you live in the same city, right? When you talk to each other, you never say like the name of the city perhaps, but just like the street name. So you assume you live in the same city, right? So that will be a relative path. So the same here. Like when you do relative path, you can say 2 dots like this and 1 level up. Remember that 2 dots means 1 level up. So 1 level up, so meaning like, for example, you want to access the file which is located 1 level up, and you say so. So what is the assumption? The assumption is that you're located in the current directory such that there is another directory above, which is called test, or like otherwise the file is located 1 level up, right? That's the assumption. So if you move somewhere else, that path will no longer work. I mean, it might, but if you move somewhere else far away, it will not work, right? So it's relative where you are located on the Unix. So absolute path will work all the time. So why people use relative path? Because it's just convenient. It's just shorter to write, right? Rather than write all the time absolute path when you're typing. It's just inconvenient. Okay, just want you to understand. Okay, so permission levels. Again, on the Google Collab probably will not do much, but I just want to kind of give you this concept so you know. So, so there is user, there is a group, and there is others, right? So like on Unix, you have user as you, your friends, meaning you're somehow the group. And it's different definitions on different machines. And then the rest of the world, others. So when you, and there is 3 types before that. So we have 3 types of what you can do with the files. You can read the files, meaning like you can look at them. You can write, meaning you can modify the files. You can add something to them, delete something from them, or like just modify them. And X means you can execute. And then execute, usually you execute some scripts or some software commands, tools, right? So you execute them. Okay, so fundamentally 3 types of things you can do with the files or directories. So
what's the difference, sorry, between read and execute? Like if you can read the file, can you just execute it?
No, I mean, when you read the file, you just look at the file, like content, right? You look inside the file and see what is inside. If you have a shell script, if you look at the shell script, it will not run, right? For example, the shell script, I don't know, is moving the files, for example, right? If you look at the shell script, you just see the code. But if you want to execute it, put it in action, in other words, you need to run it, and then it will start doing like moving the files, for example.
But if you have permission to read, you can actually have permission to execute it if you like just copy the script and make a new file, no?
No, not in Unix. I think it's done intentionally. So actually by default, by yourself, you're not able to, at least on Unix machines, or at least at Unix cluster. I don't know about Ubuntu, like your local version, but
the same.
The same? Okay. Yeah,
yeah, it's correctly for Linux.
I don't know why it's done, but I think it's on purpose. So you don't run commands accidentally, because some shell script can be devastating. For example, how about you have a shell script which deletes all the files in your system? So something.
Why
like
would you have such a shell script?
No, I mean, like I just make it dramatic, but I'm saying, you know, like some version of it. So sometimes it's dangerous. So you need to be very mindful, or like you really need, Unix wants to make sure what you do is what you mean, right? Like, did you mean to run it? So I think it's like they want you to take extra step and give permission to yourself to run it. I don't know, this is how things are. So you need to give permission to yourself to run it, and then you can run or execute, right? Okay, so if you do ls -l, you will see kind of this table or like long information about the files. So what you can see, there are three, I mean, there are like the very first column will tell you everything about the permissions you have for the file, right? About you as a user, group, and oh, sorry, it's a little bit shifted, and the rest of the world. So it should be free. So let's see. Yeah, we need to improve it. So basically, if you have the string and you count one, like the second letter, the third letter, and the fourth letter, right? R, V, and dash, right? So then R means writing, W means, I'm sorry, R means reading, W means writing, and you don't have permission, you don't have X there, you don't have permission to execute. And again, sorry, that's shifted, we need to. So, and for the group, you have R dash dash, means you have reading, the rest of the group have reading, but cannot change your file and cannot execute, and the same for the rest of the world. So, and then the very first character devoted to the type of the file. So you can see that if there is a directory, it will say D, like right here, it will say D, and here it will say dash, right? So there is nothing here. I mean, it's like a file, so it's not a directory. Okay, so that's how things are. So let's see how we change that. So we say there is a command chmod, change mode, and you can say chmod G+W, for example. You say group. I want to grant permission to the group to be able to write, meaning I want to give permission to the group to change my file. And you can say many files or just one. In this case, we said we want to change the permission to this file. So if you pay attention here, so before we run this command, if you do ls-l, the group will not have the permission to write, but afterwards it will, right? You can see W here. Okay, then you can take, if you change your mind, you can take it back. So you just say, you need to say minus, G minus V, and you take it back. Okay. Any questions so far? So again, like we'll see what we can cover again in Google Collab. So we'll be able to cover something, but like I really want you to kind of understand on the high level that this is the concept.
What's the difference between others and all?
Yeah, that's a good question. So it depends on the Unix cluster. So for example, sometimes if there is, like for example, on our cluster, I think the PI, like the professor, will be having like basically the lab, for example, right? Like one particular laboratory will be like what is called group, right? Group. It will be some group because there is a PI who's like approving people and so on, and others will be like just the rest of the world, like everybody else on the cluster, everybody on the cluster. So, and there is some difference because, for example, you want to share some files between your kind of colleagues in your laboratory, but you don't want to share with everybody at university.
All right, got it. Thanks.
Cool. So, okay, so another I think my favorite one in Unix, so it's redirection. And redirection and actually pipelines, So we'll get to that, my favorite. So redirection. So when you type something, right, like when you do ls to see what is inside the directory, you see the output on the screen, right? That's how it is. You type something, there's something going on, and you see output on your screen. Now, how about you want to save it? Somehow it's very essential information. You want to save it. So you can redirect to a file. You can redirect the output which commands produces to the file. Sure, you can. So this is what you do. Program A, redirection sign, like greater sign, and for example, file.txt. If the file, usually you want to make sure the file doesn't exist. But if it does exist, it will be overwritten, meaning you will delete everything what used to be before in the file.txt and will be overwritten by the new content. So, however, if you would like to append, meaning that you would like to add content to the end of the file rather than to overwrite it, then you can use kind of double redirection sign, like this one, right? Two greater signs. Then it will be appended. You will preserve what used to be in the file and you will append to the end. If it was nothing to preserve, you will add it just to the file. And I think when we talk about one redirection sign, like one when you're overwriting the file, I'm not aware of the way to save it, basically, except like if there is a backup on the system. But there is no kind of like warning you can get, anything like this. At least I didn't manage to do it. So that was one kind of weak point when I'm operating the Unix. Like I assume I will never make mistakes like this, right? So I will not overwrite some essential files. Okay, so let's see. So another command we will cover, actually we need to cover WC minus L. So let's hold on this, okay? We'll skip it for now because we need to know few more commands. So pipeline, that's my favorite. So you can combine, basically you can, like on the high level, sometimes you don't need new commands in Unix, but rather you would like you can combine multiple commands, and that will really gives you the power. So what is pipeline? Pipeline, you write like this. You say program A, pipe character, like the bar, and the program B, okay? And so on. You have many kind of pipe like steps in your pipeline. So how to read it? It's very simple. You just say program A's output serves as the program B input, right? So it's kind of floating, right? So whatever program A produces, it gives to B. B is taking this, process it, gives to C, and so on. So it's like information is floating in the pipeline. So what will be the analogous way to write it? There is one. So you can actually use the intermediate file, right? So for example, you run program A, you save everything what program A produces to the file, and then you kind of input from that file for program B, and so on. That's fine. What is the disadvantage of that? First of all, it's 2-line versus 1-line. I really like, since we're talking about command line, it's always, I mean, it's very convenient to have things like 1-line solution to things. And I think this is what will be like looking for, to write this 1-line solutions for Unix. So it's inconvenient, but then also you are using space, right? How about the output from program A is very large? So you're taking the space. So after you run those like 2 lines, you need to remove intermediate files, not just like too much work, right? So pipelines are nice in that sense. Okay, so one more command and we can try pipelines. So command WC, very essential one, it counts. All it does is just counts. So it counts number of lines, number of words, and number of characters in your file. So if you run WC and the file name, it will give you 3 numbers, as you can see here, number of lines, number of words, number of characters, okay? And then sometimes, I think the most often, I think personally, I'm using -L. I don't care usually about number of words or characters, not that much. I really care about number of lines. For example, somebody gives me the file and saying, can you analyze it? I'm like, what is like, like I always, and then if I want to see that the, like everything was copied, or at least it makes sense, I run WC -L and see the kind of, what is the file I'm dealing with? Is it like 10,000 lines? Is it like 10 million lines and so on, right? So that's the command I often run just to get an idea about the file. Okay. So, so WC, right? If you get the file, you can do WC and name of the file, and you will know those 3 things about the files. Again, number of lines, number of words, and number of characters. Good. Then you can do WC -L, and you will get only the number of lines in that file. Sure. Now let's do the pipeline. You do ls. You know what is ls, right? ls gives you the number of files, not the number. ls gives you the files and directories in the current directory. Sure. So when we put the pipeline and we do WC -L, so can somebody tell me what this command does?
Counts how many files are there.
Yeah, exactly. Yeah. So it counts how many files are there. Exactly. How? Because the ls, when you run ls, it will produce the output. And the output is what? Like every line is the file or directory, right? You have like some output. So all you need, it's kind of a file. So WC -L will count. And that's essentially will be number of files and directories in your current directory. right? So if I ask you to that before, for example, can you tell me the command which does that, right? Which counts number of lines, I'm sorry, which counts number of files and directory in the current directory. You don't need to know any new command, as you can see, right? All you need to do is to combine existing ones. And I think that's another beauty of Unix, that often you don't need to remember many commands, rather you need to have a logic to combine them, and you know, like, and that essentially will get the commands you need. And pipeline, again, is very, very powerful in that sense. Okay, and then in that, as you can see in that particular directory, we got like 5 files and directories. So also very important command. Like often you want to know what's going on in my directory, how many files and directories do I have? Okay, so another piece is that cat is actually the command which you can concatenate the file, meaning that you can glue them one by one, right, in the order you provide. So in this case, you say, I want to combine file large.txt and the file test.txt. Right, and and you can do that, and then basically we'll get on the screen output of the file, which is combination of both, right? Like they're glued together, combined together, like one by one. Now, if you need that file, you say, oh, that's a really nice file to have. Sure, you can save it, and you know I'm talking about second line here, right? You can save it to a new file. For example, you call the file all.txt and you use the redirection sign. Right? The redirections of this concatenation will go to a new file. Sure. You can also use some sort of pipeline. You concatenate those files and put the bar and say wc-l, which will give you the number of files, I'm sorry, which will count number of lines in both files, right? Okay, so you can see like you have combination of things you can do, right? So like now, as you're getting more and more commands, you have kind of opportunities opening for you. You can combine them. You can like, you know, like ask different questions and so on. So any questions? We'll skip this.
Yes, I do have a question. It's regarding the files we can use. So I've seen that we can use the text files. I suppose we can also use CSV files. What other files can we use? I suppose we cannot use docs files like documents or Excel files. That
That's a great question. Yeah, that's a good question. So in the Unix, I think essentially you can use TXT files, right? Like fundamentally. So what is TXT file? The file which can be like, which is the text, right? So, and then actually it doesn't matter how you call it. For example, you can call this test.txt test, I don't know, test.moldova, and it will not change anything, right? Like the Unix will not care. I mean, it will be inconvenient because like you will be confused or somebody else will be confused when you share this file, but it's fine. Unix will not care. Actually, if you do CSV file, for Unix, at least for those commands, this command will not care that there is a comma delimited things, right? It will, at least the commands we have covered so far, will not differentiate the columns because it doesn't know that there is a rule that the columns are delimited by comma. So it will treat it as normal TXT file, right? So when you talk about the doc, like the Microsoft Word, like Excel and things like that, those are not TXT files. Right? You cannot, I mean, probably you can open them as TXT, but it will be like a mess, you know, like stuff there. So they will not work because you need special program to open that. So there is, I would say that's a fundamental difference between CSV file and then Excel, for example. That CSV file, there's a very simple rule, right? Like it's a TXT file where every column is delimited by comma, right? That's the definition, that's it. So you can open that in many programs. As Excel, you can always only open in Excel and several more programs. I think that's the difference. But yeah, like anything like Excel or like any kind of complicated file formats like that, or PDF or so on, I'm not suitable here.
I'm not exactly sure, but I know that JSON is another kind of formatting file.
Yeah, but JSON is a better one. There is a more clear rule, and then there are programs which open. We'll not cover it. We'll just stick with CSV for simplicity. But for JSON, yeah, I mean, it's alternative in some sense to CSV. I'm not personally using that much. Somehow I am fine with CSV files, but I know people like it. So, but we'll not cover JSON here. But I think it's more or less. So it's a simple rules how you present information. As again, Excel is very program or whatever company specific. So it's not kind of simple at all.
Excuse me, but I did not quite get the cat command. Oh.
Yeah, so the cat all it does is concatenates. Concatenation means that you put one file and then immediately another file. So you glue them like this, one by one. That's it. That's all it does. So if you have a file, one file is 1, 2, 3, 4, 5, right? And another file is, let's say, 6, 7, 8, 9, 10, right? If you cat file number 1 and file number 2, you get a new file on your screen, which is from 1 to 10, because you combine them together, right?
Okay, but if you want to put that information, like combine them into a new file.
Yeah, this is what you do. This is the second command. You redirect to a new file. And
but
that
you
file,
don't lose,
yeah.
You don't lose the original files. The
No, you're not touching. You're not modifying at all the original files. That's a great point.
So it's kind of a copy of one file, copy of second file, and combine them into a third file.
Yeah, but specific copy, because copy is very large. Concatenation, there is a very precise term. Concatenation means like, glue them one by one, because you can copy also like this, right? Like, vertically, but we are doing, right? We are like horizontally, but we are doing kind of vertically, one by one, right?
Okay, thanks.
Sure. Any other questions?
And for the cat command, so the structure of two files must be the same.
Yeah. I mean, but we are not talking here about CSV files. We're talking about very simple files. Yeah, there is no. Yeah, I mean, to make it useful, it needs to be the same structure. But in order to command to happen, it can be anything. The cat doesn't care. It would just like glue them, and that's it, right? But yeah, I mean, actually, that's a great point, and that anticipates what we will do tomorrow in Python, right? So you will see that sometimes Unix is not enough to deal with, let's say, structured file, as you're mentioning, right? Like CSV files, because if you concatenate them, it's not easy, because, for example, think about that. You have one file with 5 columns, and you have another file with 6 columns, and you say, I want to concatenate them. So this command will not do a good job, because you need to throw away one command, for example, or you need to put missing points. So Python can do a really good job on that, and it will cover that. That's like why, that's a kind of like you're anticipating, like, why do we need Python then besides Unix?
I would like to add a plus for CSV against JSON. The CSV file are smallest, because JSON repeating are smaller.
Oh, I see.
Because JSON file, each value have the key. It's repeating for each row. So this is CSV it's better.
I see.
The information.
Oh yeah, great point. Yeah, I don't know, but what is the advantage? Why people like? Do you know why people like JSON? Like, what's the advantage?
Because it's easy to convert it to an object in JavaScript, for example. So it's like an object.
Right. So it's more like for Java, but like for Python, probably there is no point.
Yeah, it's like for programming to be easy to access the key value.
Right. Okay. Yeah. So yeah. Thanks, Mihai. That makes sense. So yeah, but we'll stick with CSV files for Python. I think they're perfectly fine. Okay. Any other questions?
Maybe global question, yes, sure. to get the general idea. So Unix, we use these commands just to organize our directories, yes, to move the files, to name, to rename, yes. But I understood that there is a possibility to save the output, yes. There are some commands, but is there any commands to have an input data, or there is no need to input data?
Usually you never input the data. I mean,
we do not input the data. We can give the link to the data, right?
Yeah, you mean like, like enter 10 digits. No, that's only like in programming exercises in school, you enter the data like this. I mean, data is large. You cannot input the data. I mean, usually, the only possibility you have like a printed something, and you need to input that. But that's, like it doesn't happen. No, it's not an effective way to input the data. It's usually data is given to you somehow through some file, right? So you're analyzing the files rather than inputting the data. I don't know if it makes sense, right? Like you're dealing with the files which are already there somehow. Like you imported that. For example, you imported that, right? Like you have a CSV file, which for example, somebody gave you Excel file, right? and you say, Excel, I don't like Excel, let me convert to CSV. And you will be working with that CSV file. But so you do it prior. I would not do it on Unix. If you need to convert from Excel to CSV, you will do it on your laptop or something like this.
And our scripts in R, in Python will be also kept, so we can have some access to these scripts using commands.
What do you mean access? Like you will write them yourself, so you will have access to them.
Yes, we'll have an access to the scripts.
Yes. Yeah, you will have access to the scripts, and then you can use them as the new data comes, right? So like, right, like you have access to the scripts which you can use to analyze the data, like whatever the data it is. Cool. Great. Any other questions? All right. So let's see. So let's see. We'll kind of glance through the rest of the slides and let's see what is necessary. So okay. So I would say let's cover grep. I think it's powerful, but that will be probably will not cover much more than the grep. So so grep is, I think it's powerful. So basically what it does is search for something, right? It searches for certain strings, right? For example, you have a big file and you would like to identify the lines which have the word apple. So you can do that by grep. So then there are several options, right? So for example, you can find the lines not containing the word apple, -v. You can count the number of lines which contain the word apple, -n. The number of line numbers, I'm sorry. So -n will tell you the line numbers where the word apple occurs. -c will count number of lines which has the word apple. Then you can say -l, which give you also the file names, because you can search for the multiple files. And then you can make that search case insensitive, meaning you don't care about capital letter, not capital letter, right? For example, if you think somebody did the mistake pronouncing your first name or last name and forgot to put capital letter, like you can kind of still find yourself by doing -i. Okay, so let's see some examples. So if we have this file, large.txt, where basically there are multiple lines, multiple numbers in that file, so you can say grep 1 and then find the file, the lines which contain the digit 1 in this case. So it will be 1, 10, and so on, 19, right? Those are, every line here will have a digit 1. Then we do grep -n. So we also see the line number. So in this case, they are the same because, we created this file artificially. So it's like 1, 2, 3, and so on. So line number is exactly the same as the content of the line. So -c will give you number of lines where digit 1 occurs. -l will just give you the file name where digit 1 occurs. For example, we are looking through two files, large.txt and test.txt, and only in large.txt the 1 occurs. So that's why we see large.txt, but it doesn't occur in test.txt. That's why we don't see it. So, but if you don't use -l option here, you can do grep 1, right? Two files, and you will see both. You see the file name and the line itself. Okay. So, any questions? So then, if no questions, I have a question for you. So what will be the alternative way to do this command? Grep -c 1 large. For example, what about you don't remember -c, just forgot. All you know is grep. Can you write the command so that you can still count?
grep 1 pipeline vc
-l.
-l, yeah.
Yeah, exactly. Yeah, it's wc. So wc -l, exactly. Yeah. So basically, sometimes you don't need to remember exact the commands, but rather have like the logic, and then just use some alternative. I think exactly that's absolutely the valid way to do it. Okay, so let's see. We will see. So I'm just thinking, should we go for this or take a break? Regular expression. Yeah, let's maybe take a break. So let's take how much? So let's come back like in 12 minutes. So basically when the TAs will join. So and then I think we'll put some content of Unix. We'll work more on this tomorrow. So and then I want you to guys have plenty of time to kind of go through the commands and so on. So okay, let's take 12 minutes, 11 now already, 11 minutes break, and we'll be back. We talked about grep. We'll talk about this. Okay, so actually like one motivation why the grep is not enough and kind of why you need Python and other things will be the following, right? So there is different formats in bioinformatics. For example, the format called GTF, it's about gene expression, not gene expression, gene annotation data. So basically you know where its exons are, introns are, and so on. So this is one line, right? It says chromosome 21, HD 19, that's a reference genome, the build. And then it says coding region and then coordinates, like in the genome, right? So like big numbers, just coordinates in your genome, where that gene is located. And gene ID, the ID of the gene, and transcript ID. And then, okay. So, and then you say, will the grep be enough, right? If you know grep, can you extract, for example, lines? So you have a huge file, so obviously across all chromosomes, right? From one all the way. So, but you say, I don't really care about all chromosome. I would like to modify that file so that the file will only contain chromosome 2. And what you say, hey, I know grep. Grep is a great command. I like it a lot. Let me try it. So you're running this command. Oh, let me make full screen. See maybe better. Yeah. So you're running this command: grep chromosome 2 HD19.gtf, right? And you think it will work. So what you're doing essentially, you're asking the question. Grep is asking the question: does that line contains string chromosome 2? Yes, no. Yes, then it will output that. And then you redirect to a file which you call chromosome2.txt. Okay. Now you say, I would expect only the lines from chromosome 2 will be saved to that file, right? But will it be the case? So is anybody seeing any issues with that approach?
Well, you'll only collect the first line of chromosome 2, where it's the title said chromosome 2, and you also collect the chromosome 21. So
right. So exactly. So like the problem is that when you say I would like to have chromosome 2, you're talking about the string, right? Like a string of letters. The computer will not differentiate chromosome 2 and 21. So what will happen, actually, if you look at carefully at the chromosome 21, you can see that chromosome 2 is inside as the string, right? CHR2 is inside. So computer will think, oh, that's chromosome 2. So it will take it, right? So you can see that you probably need something more than the grep, right? Grep is powerful to the certain extent, but this is one motivation for Python. So probably to analyze this file, Python will be a better idea. Okay, so we'll skip regular expression because that will be probably too much. And then let's see. So we're skipping this part. And then probably, did you talk with Cynthia about SID and Oak? Probably not, right? Yeah, I don't think so.
No.
No, that's fine. Then it's like, let's not, that will be too much because Python is kind of doing that. So we'll remove those slides. We'll send you updated version. Yeah, and then so we'll talk about the scripts, right? Kind of like you talked enough during the TA session, but I want to kind of like tell you again on the high level. And that will be it for Unix. I don't think we need more. Okay, so and then we'll update the slides so you will not be confused with extra stuff. Okay, so why do we need scripts, right? Like we talked, like you notice, like we do for loops, we do shell scripts. Like, what's the point, right? Like, why are we doing that? So the idea is that very often we do the same tasks, right? For example, you do a lot of commands. You know, like you do something, you clean the data, you combine the data, So you have like, I don't know, let's say 50 commands. It's a very laborious process. And you did it once for one data set. And you say, okay, that's great. And you got another data set similar to this one. And you say, okay, I will do those 50 commands again. And you do them again. But then at some point you say, okay, you know what? Why don't I write the script about that, right? Like, why don't I put all those commands into the program so that I can utilize this program just like by running it rather than typing those 50 commands? And so that's the idea about the script, right? And the same for loop. You know, like if you have one file, that's great. But if you have 10 files, yeah, you can do 10 files, repeating the commands for 10 files. But as soon as you have 1,000 files, that's it. You don't have a choice. You need to have a way to look at each file automatically rather than manually, right? It's all about manual versus automatic. So we'd like to automate things. And then that's very common, you know, like probably for the very first time when you do exploratory analysis, I would say it's totally fine to do it manually, you know, command by command, because you want to learn. But as soon as you did it once, there is no point to do it twice, you know, like the same thing. There is a point of spending time and putting those commands into the script so that, you know, like, so that like you can use it very efficiently the next data comes, right? And that's very typical. You know, we are very often dealing with similar data sets. Okay. So you already wrote your first shell script, and then, so just to highlight, I think here, so what you can see in the script, you actually don't need to know anything new, right? Like, I mean, sure, you've learned for loop, but typically you can survive with what you know, right? And again, there is very, very many ways to do Unix and to write scripts. And then I think one thing you need to understand is that I think it's about being able to do it. In a sense, in bioinformatics, I mean, we care about beautiful code, but less so. And I think it's actually very interesting. People who care about the beautiful code too much are less successful in bioinformatics. And the reason is we are very practical. If things are working, maybe with the ugly code, that's fine. You know, like as soon as it's accurate and efficient, you know, like so don't kind of overstress the beauty of the code, but I think the beauty is really in the data analysis, right? So if you can do things, you know, with 50 lines versus 20 lines, that's fine. But, you know, like, so, but I think as you advance, sure, you know, like be curious and learn more things. But for now, be happy if your script is working regardless how it's written. So here, so what you can see, we are using the commands which are very much, we know, except echo, right? So like echo is just to print something. So we say my first script, you just print the message. So, and then you create the directories, right here, mkdir, right? So you, instead of typing in the command line, right, you just putting the commands inside the script, right? That's the point. And here we actually have a solution for the grep to extract chromosome 2. So the solution is the following. So we say, remember, right? If we say just grab chromosome 2, it will not work because it will pick up chromosome 21. Not a great idea. So actually we can make, and then that's fine to make that mistake, kind of, right? Like when you run chromosome 2, and then it's okay to start like that. But then what you should do, you should explore your output. You should look very, very careful at your output and catch the error. So the point is, it's not the big deal that you make a mistake. It's very important to catch it. Everybody does mistakes, but the point is to catch it, to kind of self-assess. That's crucial. So if you're kind of like lazy in that sense, you just write the commands, you assume it works. You should never assume your command work, especially from the beginning, like never. Like question yourself. Basically think about that you did a mistake and just like you need to find it. So if you have this approach, you will catch the errors because you have limited information or like limited understanding yet. So limited experience yet. So for example, here you run it. Let's say you run without /s, right? Just chromosome 2. So, and then your file will have chromosome 21. So you need to open the file and look. So if you look at the beginning of the file by head, and you look at the end of the file by tail, you will notice that you have chromosome 21, and you will say, oh, wait a minute, why chromosome 21 is there, right? And you will catch there, and you will think, okay, I need something better. So something better, and I mean, again, we'll do that something similar in the Python, but here with the grep, something better is to make a very, very simple observation. The observation is that after the chromosome 2, there is a space, right? Because it's columns delimited by tab. And in chromosome 21, and then in chromosome 2, which is substring of chromosome 21, there is 1, not the space. That's basically it. That observation, at least in this particular case, will be enough to differentiate chromosome 2 and 21. And then your code will work. And then actually there is a special character for space, /s, right here. That's a special character for space. So that will be enough to differentiate between chromosome 2 and 21. So now, when you're writing the script, you can you can hard code the files, right? Like if the files are given to you, remember you have relative and absolute path, so you can kind of like say that, right? Like you can say absolute path, but then the script becomes less useful because as you get new data, you expect the file to have a different name. So what you can do, you can say what is called command line argument, right? The command line argument, that's the way to communicate with the script. So in a way that you will tell the script, what is the file you as a script need to process, right? So so that's a good like approach. And then let's see, okay, so when you run it, so that's a command line argument, right? So this one. So when you run it, that's your script, and this is how you run it. And remember, you don't have permission to yourself to execute the script, so you need to grant the permission typically on Unix to execute the script. So that's your script. You just call it script.sh. So this is how you run it, / name of the script, script.sh. Now, a way to communicate with the script is to provide command line argument. So the command line argument, this is some string which you say after space. So basically you say the name of the script, space, right here, and first command line argument. You can put the space and have a, yeah. So like you can put a space and have a different one here. So how the script knows? Very simple. It will say, I will look at that string here, and I will look at the spaces. So whatever is in between the spaces, that's my first command line argument. So I will assess that by $1. So $1, when you run the script, $1 becomes hd19.gtf. And so on. $2 will become this one, whatever you provide in what is violet color. Right. So that's how you communicate with the script. The reason, again, you want to make script very flexible, universal, right? Like as you get new data, it can actually work, right, for the new data, rather than you don't want to go and change the script, because that's a possibility, right? You write one script, which is you say exactly, instead of $1 everywhere, you say exactly the name of the file. But that's not convenient. As you run next time, you need to go inside, change it. That's a bad practice. That's not a good idea to do that. So it's a good idea to use command line arguments. Okay, so I think that's it. We need.
And,
yeah.
Sorry, command line arguments can be for whatever you want, or only $1.
No, I mean that's a syntax, right? Like to tell Unix or Bash in this case, to tell Bash that you want command line argument number 1, that's $1. If you want command line argument 2, $2. That's just the syntax.
What would it change if we said $2?
Then
would it change anything in this code?
In this code?
Yes.
Wait, so if you change you're asking if you do change the code and put yes, if you change the code, then you need to run it differently. When you run it, you need to provide second command line argument, which is like what, violet or whatever, blue, I don't know what it is, violet color here, right? You need to say if you would not have like if you run, okay, how about this? If you change $2 here, right? And then you run like this, the script will fail. Why?
Yeah.
Because you don't have second command line argument. So what you need to do, you need to provide second command line argument. Then you're fine, right?
But why would we want you say to run only the first line argument? Like if we had 10 files, it will only run the first one.
Yeah. But in this case, we have one file. This script processes just one file.
Okay, got it.
Right?
Thanks.
Yeah, like the script will, actually what it will do, if you look carefully here, it will split, it will create 3 directories, right? You can read the code already, right? If you look carefully here, you will notice that mkdir creates the directory. So we just create 3 different directories, right? That's it. And what we do, each line of grep will select the lines corresponding to chromosome 2, chromosome 3, chromosome 21, and put them into distinct files. As you can see here, it will put inside those directories, like right here, right? So that's all the script does. So the script is working for a single GTF file, right? So we don't need 2 command line arguments in this case. The script does distinct task, you know, like, and then of course, if you want to run that script across 1,000 files, you would write for loop on the top. For
everyone that has the termination.
But on the top, but not inside. I would say it's not, my preference is not to put the for loop inside, but rather outside. I would rather have script to work for this one file and do whatever I want, because I still can use it for a single file. I want to have that opportunity. But if I want a for loop, you can write the wrapper around that and put the for loop, right?
Okay, thanks.
I mean, it's a matter of like choice, I think, but I think it's like you want to define script as distinct thing, like it should do one thing. Like in this case, split one file into whatever small files, and that's it. That's it. It's okay to have that short script, but it's better to have some so you can understand what the script does, right? Of course, you can write the scripts which does the entire analysis, but I would argue that's not a good idea. It's better to split into the scripts which you understand what they do. It's easier to understand, like kind of similar task, you know, like one complete task. Okay, so I think that's pretty much all, you know, like we need to know for Unix, at least for now. I mean, you will practice Unix more when you do like the, what is called your individual project as well as bioinformatics class, but like that's, I think it's a good start. And again, if you understand something, that's great. If you don't understand something, watch the video again, try to type it yourself again. And if you got the error, make a screenshot, ask the TA, or like send on the Slack channel, or like personal message. You don't want to see anybody to see it, that's fine. It's like send this personal message, I guess, or like email. Okay, that's fine. Great. But I think it's like, again, don't be ashamed if you like, there is no bad questions, you know, like I think what you are experiencing, like if you're getting the error, probably another 20 people or 10 people getting the same error, right? So it's always beneficial to bring it to TA and say, hey, I got this error, can I share my screen? And like, you know, like, and you can help me, and they will help you. Okay, so that's the same story, right? Like we want to get you accumulate kind of enough knowledge so that you can be independent, right? Like you can open this, like as it was mentioned, there is a lot of resources on the web, you know, like you can open Google or Stack Overflow, whatever the resource, and you can read it and understand that more or less, right? So you can utilize, that's your goal, the goal to understand enough so you can understand that, right? Because now I think Unix, I hope you will understand certain commands, and we saw, right? Like I gave you the example about ls. You say how to sort by date, and you say ls. So if you do it now, I imagine you will understand what they're talking about, which is great, you know, like, so that's how you will build your knowledge. Okay, so I think we're done with module 1. So any questions about module 1?